All right, everyone, welcome to Chapter 3. Chapter 3 is going to be about the cell. So we talked about chemistry and chemicals in Chapter 2. And what we're going to do is we're going to put all those chemicals together and eventually make more complex structures that exist in the cell. And then eventually cells by themselves. And then we'll build into tissues and organs down the road. So let's get started. Once again, here's some learning outcomes for you. The reason why they don't start on number 1 is 1 through 32 is coming from chapter 1 and chapter 2. So this is just a continuation for this first module for your first exam. So by the end of the chapter, please make sure that you go back and understand and be able to identify and answer these statements and questions. A few more learning outcomes for you. And a few more learning outcomes for you. These last two you actually already know. We covered this in chapter 2 in regards to DNA and RNA sequencing. And all it's asking you is use the below DNA sequence to write out a complementary, or the other side of the DNA sequence. So all it's asking for are the best friends. So you already know that A is always best friends with T, and G is always best friends with C, and C best friends with G, and so on and so forth. And for number 50, it's the same exact thing with RNA, except that there's no T, right? What do we substitute for RNA? And very good, your answer should be U, uracil. So A is best friends with U, and then we'll have a C, we'll have a G, we'll have an A, that kind of stuff. So you actually already know those last two. Awesome. All right, let's discuss cells. So cells are the smallest functional unit in the body. Another way of saying that, they are the smallest living thing in the body. And remember the 6 criteria that we talked about for life. We talked about metabolism, we talked about responsiveness, we talked about movement, we talked about reproduction, we talked about growth, all those things. You have to display all those characteristics in order to be alive. So things like oxygen and carbon dioxide and our sodium chloride, all those things are carbs and proteins. All those things we discussed last chapter in chapter 2 don't display all of those. Those are just a bunch of combinations of atoms and molecules and compounds. But with a cell is complex enough. We have enough of these molecules and compounds where we actually can display those 6 criteria for living. Cells can metabolize. Cells will build things. We'll talk about what they can build. We'll talk about what they can break down. Cells can reproduce. They can make more cells, right? Thankfully, your skin makes new cells all the time. Your stomach lining, your bone cells. We're constantly making new cells from our old cells. Cells can grow, right? Hopefully when you go to the gym, your muscles grow. And when we get older, our organs grow to be proportionate with the rest of our body. So cells are the living units of our body. They're the smallest living units in our body. And they can vary greatly. We're going to see all sorts of different crazy cells. We're going to put them together in tissues next chapter, but we're going to have weird pancake-looking cells. We're going to have square and rectangle cells. We're going to have weird cylinder-looking things, alien-looking structures, and big bubble-looking cells. And all of those different shapes are going to be responsible for different functions, right? Big key theme for this entire class, this entire curriculum. Shape determines function. And some of these are really, really, really small, and some of them are just really small. But all cells, for the most part, are pretty microscopic. So as you can see, many, many different shapes and sizes and categories of cells. You can see a muscle cell, you can see a fat cell, looks like just kind of a big bubble. You can see a specific type of bone cell, kind of looks like a weird octopus with more than 8 legs. Your red blood cells kind of look like these little donut things. On the bottom, your nervous cells looks like this little alien spider leg looking thing. Your epithelial cells are just these weird shapes that are stacked on top of each other. And once again, all of these shapes are going to have very different functions because they are shaped differently. First things first, in regarding the cell, 99% of the cells in your body have these three things. They have to have these three things, otherwise they are not a cell. Number one, and in no particular order, but number one is a cell membrane. I'm just going to put cell M. You can see the spelling down here, cell membrane. And that's going to be our first topic of discussion with this chapter. Number two, some of you have heard this and seen this, a nucleus. 99% of the cells in your body, or more than 99% of the cells in your body, have a nucleus. And then everything inside of the cell membrane that's not considered the nucleus is floating in this kind of water jelly-like substance called the cytoplasm. So when you see all these different terms we're going to be talking about, like the mitochondrion, the Golgi apparatus, the endoplasmic reticulum, the lysosomes, all this crazy stuff, if it's not named nucleus, it belongs in the cytoplasm. So the nucleus is kind of its own thing. And I'll show you a picture in just a moment of what these three structures look like, but almost every single cell in your body has these three things. The weird exception would be your red blood cells. And even actually, your red blood cells are made with a nucleus. It's just once they get older and more mature, they spit out the nucleus to optimize their function. We'll talk about that down the road, excuse me. But for the majority and for our conversation in this chapter, all cells have these three parts. With cell membranes, there are two basic types. We're really only going to be talking about this first one, the plasma membrane. That's going to be by far the majority of our conversation today. But I do want to introduce both of them. So the plasma membrane first, this is what we discussed in Chapter 2 when we were talking about those phospholipids, specifically that phospholipid bilayer. What we have is we have a phosphorus head that's hydrophilic, that loves water, and it has two fatty acid tails, and then we have another layer of that, and they're kind of inverted like that. So what we have are the fatty acids, the tails, facing in towards one another. And once again, that's really, really important because water and fat don't get along. So we're creating this kind of barrier to separate the water environment from the inside of the cell to the outside of the cell and vice versa. The other type of membrane that does exist in cells are the organelle membranes. So certain organelles are the little mini organs all throughout the cells located in the cytoplasm. So basically everything that's not called a nucleus. These also, some of them can have membranes. And that's going to be really important just for separating things and compartmentalizing things. Remember, one of our big life requirements is to be able to separate and create boundaries. So organelles such as mitochondria, as you can see in the picture on the bottom right, some endoplasmic reticulum, Golgi apparatus, and then some specialized bubble-looking things such as a lysosome and peroxisome. They also have their own unique membranes that help with their function. But for the most part, and for the immediate future, we're going to be discussing the plasma membrane on the next several slides. When learning about what the phospholipid bilayer or the cell membrane is all about, a really great way of explaining it is something called the fluid mosaic model. And what a mosaic is it's a picture or a pattern with many different parts to it. Thing that comes to my mind is if you look at like tile, as long as it's not like same uniform color like a tile in a kitchen or something. You have a pattern where it's all kind of looking the same color, but you have all these like splotches and little spots and all these cool little designs all throughout the tile. That's what a mosaic is. So with the outside of our cells, with our cell membrane, we have something called the fluid mosaic, which means these things are constantly moving. And as you can see in the picture, I know it's a little small, but if you take a look, all the things in red are those phospholipids that we've been talking about in the chemistry chapter and what I just previously drew. But notice that there are some other colors inside of this phospholipid bilayer or this cell membrane. We have these little blue things, we kind of have these little yellow things interspersed between them, have these green things kind of sticking out on top. And those are all sorts of different proteins and fats and carbohydrates. And all of these things are constantly moving. They are not stationary. In fact, these the phospholipids move millions of times per second. They are constantly in motion. And one of my mentors explained it really, really well to me as I was trying to grasp this concept. Is he said, picture a bathtub full of water, and you just dumped like 300 ping pong balls in the water. And even if you're not stirring the water or moving the water or putting more water in, those ping pong balls are constantly moving, right? They're constantly hitting each other, bouncing into each other, bouncing against the walls of the bathtub, changing their direction, their orientation. They're constantly moving. And that's kind of what's happening on the outside of your cells. So it's a very changing environment. We're going to be getting things into the cells, such as nutrients. We're going to be getting waste products out of the cell, so stuff that needs to be constantly coming in and out, and we're going to have a lot of moving parts with this cell membrane. So we call it the fluid mosaic model, just constantly changing. All right, we've already covered a big portion of this slide. Really good picture in the top left corner. You can see what we're talking about with the phospholipid bilayer. Once again, a phospholipid is just one of these red head-looking things with the two fatty tails. But since we have two layers, 1, 2, we call it a bilayer. And what we're talking about, big picture, everyone, is this is the outside of your cell. This is the wall of your cell, if you will. So here where you see this word extracellular, that means outside of the cell. And intracellular would mean inside of the cell. And once again, the majority of the inside of your cells are water. The majority of the outside of your cells, also water. So we have water inside, water outside. That fat barrier, those fatty acid tails facing in towards one another, creates this nice, happy barrier so our cells don't get disrupted. We don't swell up with too much fluid or dehydrate too quickly. So we have a hydrophilic and a hydrophobic component to this phospholipid bilayer. What I'm circling in green right now are the phosphorus heads, and those structures are actually touching the water. So since they're touching the water, they don't really have a big problem with water. They actually like the water, so we call that hydrophilia or hydrophilic, as we discussed last chapter. And you can see, if you want to actually look at the molecular structure of a phosphorus head, you can see it's a phosphate group with a glycerol molecule, that sugar that connects to those fatty acids we talked about. And then the stuff that I will be circling in black, that is the hydrophobic part. Those are the fatty acid tails that do not like the water. Once again, phobia, don't like, afraid of, that kind of thing. So, what stabilizes this? What keeps us, with so many moving parts, with this fluid mosaic model constantly moving back and forth and shifting and changing shape, we need something to kind of keep this intact, keep it organized, make sure that we're not just falling apart, right? And we briefly discussed this last chapter. What actually helps this fluid mosaic model and this plasma membrane stay stabilized is a very, very important lipid called cholesterol. Remember, we talked about the importance of cholesterol in the diet. This is probably the number one reason, is because your billions and billions of cells need cholesterol in order for the plasma membrane to be healthy. So what cholesterol specifically does is it helps maintain the fluidity, or that fluid mosaic model, with temperature changes. And the way I kind of always pictured this in my head as a student was with saturated and unsaturated fats. We kind of need a balance, right? We don't want the cell to be too liquidy, right? We don't want the outside of the cell to be like olive oil, because then there won't be any shape. If the outside of your cell is just liquid, it's going to lose its shape, and if a cell loses its shape, it loses its function. But it also can't be saturated fat either. It can't just be solid animal fat from like a steak, because then it's just a solid mass and nothing can get in or out. So oxygen and nutrients and good things can't get into the cell because of that big block wall, and the bad stuff that builds up in the cell, like carbon dioxide, can't get out. So then the cell's going to become toxic and starved, and it'll die. So we kind of need this weird balance between not too solid, not too liquid, kind of just right in between. And cholesterol actually helps us accomplish that. So very, very important in regards to stability. And then, as you saw in the previous picture, we're going to have all sorts of cool proteins interspersed and dispersed, that's what I'm trying to say, throughout the plasma membrane. And we talked about some of the cool functions that proteins have. We'll recap some of those in the next several slides. Nice little recap here. Remember that there are several functions of proteins. We talked about enzymes and antibodies and all sorts of cool things. One of them was proteins help us transport things. And you can see with this picture that we have a phospholipid bilayer, and this big blue thing kind of right in between, or smack dab in the middle of this phospholipid bilayer, is called a protein. And this protein is helping things get in and out. So say that these things are more water-like, they're water soluble, so they don't mix well with fat. So because they don't mix well with fat, they can't get in and out of the cell. So we need kind of a secret passageway or a detour route, and some of these proteins can help us with that function, or with that goal. So as we just discussed on the previous slide, proteins can kind of be their one-man show and just do their own thing, but they can also combine with sugars and fats and have really neat specific functions. And it's not just proteins too. Sugars and fats can get together by themselves and do some really neat things. So when these different molecules, carbs, fats, proteins, combine, they come up with these really cool names, and they have a lot of neat different things that they're good at. Glycoproteins and glycolipids, I'll talk about in just a second. You can see a lot of their functions over here. Very similar. What a glycoprotein is simply a sugar attached to a protein, and obviously a glycolipid would be a sugar attached to a lipid. But lipoproteins are really important. This is what actually helps transport cholesterol throughout your body. So once again, I mean, yeah, if you have bad cholesterol or too much of bad cholesterol in your body, that's not a good thing. But you do need cholesterol. We just talked about the importance of cholesterol stabilizing that cell membrane. And you need that in all your cells, right? You have cells all throughout your body. You have cells in your brain, you have cells in your fingers, cells in your kidneys and spleen, you have cells in your ankle. So we have to transport this healthy, good cholesterol all throughout the body to all of your billions and trillions of cells. And how we do that is we need a taxi driver or an Uber driver, and those Uber drivers are called lipoproteins that kind of escort them throughout the body, throughout the bloodstream. Skipping down towards the bottom, proteoglycans. That's a protein mixed with a sugar, and these are really neat. They like to combine with collagen. Remember, collagen is a protein, so it's kind of a protein sugar combining with another protein. And this is kind of the basis for your cartilage. throughout your body that helps provide lubrication in the joints and protection of your skeletal joints and other tissues. So when we get to glycoproteins and glycolipids, as I mentioned, a lot of them are kind of interspersed. They kind of all do the same thing. Glycolipids are going to be involved with the immune response and blood types and helping cells talk with one another. But so will glycoproteins. So you can see all of these different things that they're responsible for. We'll learn about the immune response and blood types next semester. Organ transplant acceptance or rejection, whether the body thinks something is okay or whether it's foreign. If the body thinks it's foreign, we're going to mount an attack. We're going to announce war on that foreign body and try to kill it. So a lot of that is basically these glycoproteins. These glycoproteins are kind of like little satellite dishes, little receptors on every single cell in your body. And what we have are these really cool white blood cells, these really cool soldier kind of cells. And they just go around and they pull people over, pull people to the side, and they check their license and registration, if you will. And if they have a valid license and registration, meaning that I'm part of you, we're on the same team, they let you go, no big deal. And if you don't have a valid license or registration, or you are foreign, such as a bacterium or a virus or a fungus, we mark you and they kill you quickly in a non-compromised, immunocompromised person. So these kind of work as our communicators, basically like a big cell phone tower, I guess you can say. All the cells in our body need to be on the same page. They need to be on the same team. So we'll talk more about that, like I said, next semester with A&P 2. This slide is showing you an additional feature of proteins that we actually did not discuss last chapter, in chapter 2. Proteins can also act as receptors. And kind of like those satellite dishes, those cell towers that I was just mentioning on the previous slide, this is how a lot of the cells can talk with one another and how a lot of chemicals and hormones can affect cells inside the body. And a great example I like to use here is this is how drugs work. So for example, say that I sprained my knee. Say that I was walking on the sidewalk, not paying attention, I stepped funny and I twisted my knee. So my knee's really hurting, and my friend says, well, take an ibuprofen. It'll help with the inflammation, it'll help with your pain. And I say, okay, cool. So I take the ibuprofen, I put it in my mouth, I swallow it, and it goes down to my stomach. Okay, so that thing is just a really, really tiny pill. How on earth does it know to go to my knee? Because it's in my stomach right now, mixing with all those stomach acids and hopefully some food I ate. How does it know not to go to my brain or my heart or my liver or my right knee? I sprained my left knee. What if it goes to my right knee or my ankle? How does it know where to go? And the answer to that is it really doesn't know where to go. All it's looking for is the proper receptor. And that's the difference between ibuprofen and maybe like a steroid. They have different mechanisms to where they seek out different receptors in the body. So that ibuprofen is absorbed in my bloodstream, and it goes all over the bloodstream. It goes everywhere, hands, toes, head, everywhere. But it's only going to interact with an area that has the receptor for inflammation. So because I sprained my knee cells in my ligament and muscles and tendons, they are going to scream inflammation, there's something wrong, come help us, and that medication is going to be able to bind to those receptors, which will cause all sorts of neat and cool domino effects inside the cell, which will help bring down inflammation and hopefully bring down the pain. Now, I'm not saying take painkillers or anything like that. I'm just saying that's how medications and pharmaceuticals are designed to work. They talk with the receptors of the cells. We mentioned in chapter 2 that proteins can also work as enzymes. The example we used was lactase, right, with lactose intolerance or just eating milk, yogurt, and cheese. This picture right here we'll get to more next semester, but this is showing you how our bodies are really, really good at making ATP. So Once we get to the mitochondria, we're going to give you some buzzwords. We're going to say the mitochondria is the powerhouse of the cell, or the mitochondria are the energy factories or the energy makers, and then we're going to kind of leave it at that and not really discuss it in too much more detail until next semester. But how the mitochondria is so efficient, how the mitochondria is so good at making ATP, it's due to these guys. It's due to enzymes. The enzymes, remember, what an enzyme is, it speeds up a chemical reaction. It makes processes easier. And inside of your inner mitochondria, we have a boatload of enzymes. So one enzyme makes the thing go faster and helps save us time and energy and efficiency. And then we send it to another enzyme and another enzyme and another enzyme. And you can see all the enzymes here in this picture. And at the end of the day, what that helps us with is making a lot of ATP. And one more final neat thing that proteins can do is they can serve as binding proteins. And some proteins serve more in line with structural support, where they can be bound to components of something called the cytoskeleton, which is just a whole bunch of proteins that kind of maintain the shape of the cell. And this helps maintain its function, right? Shape determines function. And then other proteins can just kind of be help connect with other cells. And by holding these cells together, remember, if you put a whole bunch of cells together and they all have a common purpose, common function, we call them a tissue. And then you put a bunch of tissues together, we get our organs, right? So in order to make these tissues, a lot of these cells kind of have to be glued together. And how these cells are actually glued or held together is through these proteins. And they allow the cells to actually also talk with one another. In this picture on the right, you can actually see what we're looking at, believe it or not, is heart muscle. It's cardiac muscle. And the cells are all linked together. They're all connected. But there's these little tiny gaps, these little tiny channels that allow those electrolytes, the sodium and the calcium and the potassium, to move back and forth, which allows your heart cells to be basically sparked by electricity and for them to contract. So the proteins can bind things together, and they can also help with movement and contraction as well. Now that we have discussed the plasma membrane, we've talked about the phospholipid bilayer, the fluid mosaic model, we've talked about proteins and sugars and other stuff that's inside and connected to the plasma membrane. Now that we have that foundation, we can talk about how we get things in and out of the cell, right? We want to get good things in, we want to get the bad things out. So there are two ways of accomplishing this, and what we call this is transport, right? Just if you're transporting to school or to work, you're going from point A to point B. So there are two types of transport. Number one is called passive transport, and the second one is called active transport. For the next several slides, we're going to focus just on passive, but I do want to introduce both here just so we have an idea, so it's not completely new when we get there. Passive transport happens naturally. It kind of, if you will, obeys the laws of the universe, or the laws of science, if you will. Energy is not needed. So let me draw you a quick diagram, and you can kind of see what I'm talking about. And you already know how this works. You know the answer to this, even if you don't think you know. So what I just drew was a beaker, or just a box, doesn't matter. And that box has this wall, or this barrier in the middle, right? And it doesn't matter what we have inside the beaker. It can be a solid, liquid, gas, whatever. Let's just put some gas molecules. So on the left side, I just put 4 gas molecules. And then on the right side, I have, I don't know, let's say 14 gas molecules. So there is an imbalance. These things are not equal, right? There are a lot more on the right side of the picture compared to the left side. Now, whenever we have an imbalance, or whenever we have something that's not completely even, what we call that is a concentration gradient. Concentration gradient means that one side has more than the other. Now, even if you've never taken this class, if you've never taken a college class, if you've never taken a high school science class, it doesn't matter. You probably already know the answer to this. What do you think would happen to these gas particles, these molecules, if I erase that red wall, that barrier in the middle? Boom. What do you think would happen? Do you think everything just stays the same? And what you're probably saying at home or wherever you are is all of this higher number, this group of 14, are probably going to go from this right side of the screen to the left side of the screen, and they're all going to kind of mix in with one another. And that's exactly what would happen. So at the end of the day, once you give it a few seconds, few minutes, few hours, whatever, what's going to happen is we're going to have a beaker, and we're going to have those 18 particles all evenly dispersed throughout the entire beaker or container. So a concentration gradient, once again, is the difference, the imbalance. And what happens during passive transport is we always go from the high number to the low number. And that's what we saw. That 14 number of particles on the right moved to the 4 particles on the left to mix in and make it more even 50-50. Now, all I did was remove the wall. Nothing else happened. I didn't have to blow on the glass particles. I didn't have to heat them up or anything like that. I didn't have to stir it. It happened naturally. So there was no energy needed, and it was going to happen whether I did anything or not. For example, put some creamer in your coffee, or put some salt in your water bottle. Yeah, you can mix it, you can shake it or swirl it with a spoon, but even if you don't do those things, eventually that stuff will mix in. It could take several minutes, it could take a few hours, but those particles will disperse by themselves from that high concentration to the low areas, so where eventually all of that salt or creamer or whatever we're talking about, well, creamer might be a bad idea because it's fat, bad example, but let's say salt or sugar or even food coloring, that is always going to disperse evenly without any extra energy needed. Now, if you put extra energy, if you shake it really violently, yeah, you're just going to speed up that process. But it happens naturally. One more quick example, and then we'll move on to active transport, is oxygen. If you think about breathing every few seconds, what is happening? What is going on? What's happening is you have, right before you take a breath in, you have more oxygen or a greater pressure of oxygen molecules outside of your lungs compared to inside. So what happens is, as I'm trying to erase all this, so let's just draw a box or a circle. That'll be our lung, L for lung. What happens is, as we take a deep breath in, all of those oxygen molecules naturally rush in to the lungs from a high concentration to a low. Well, guess what? Now we have all this air filling up in the lungs, and we flip the concentration gradient. So now there's more air, there's more pressure in the lungs inside compared to the outside environment. So then what do we do immediately after we take a breath in? Well, we take a breath out. So it goes down its concentration gradient. As soon as that pressure, that air, it has an unbalance or an imbalance, then they go back to high to low. And we constantly do that every few seconds to maintain our oxygen and carbon dioxide needs. So always high to low in regards to passive transport. Now, active transport, kind of the opposite. A little harder to grasp sometimes, but this is going to be really, really cool, especially when we tie it all together with the nervous system and the muscle later in the semester. Now, sometimes in the body, not all the time, but rarely, Let's make that box a little better. So I'm going to draw my container or my beaker again, and I'm going to draw my barrier, my wall. And then here are my molecules, 3, 4, and then we'll just have a lot here. Okay, so once again, we have a concentration gradient. Do you agree? So we have a lot more on the right compared to the left. Now, sometimes in the body, what we like to do is we like to take some of these little things and we like to push it to the higher side, which increases the concentration gradient. It makes the unbalance or the imbalance worse. Now, if that's kind of confusing you and boggling your mind a little bit, good. That should be weird. That shouldn't really make sense. So in order to do that, because it doesn't make sense, because we're going against the concentration gradient, or what we say up the concentration gradient, we have to have a little energy. We have to use a little oomph to get that over that barrier. And that energy molecule that we use, you should already know the name of it. Remember our currency, the US dollar for every single thing that we do, is called adenosine triphosphate, ATP. So in order to go against the grain, in order to go from low to high in this case, we have to use ATP to accomplish that. And that is the definition of active transport. So passive transport is high to low, happens naturally. Active transport is low to high, where we have to use a little energy to accomplish that low to high. We're making the concentration gradient more unbalanced or more drastic. So for the next few slides, let's talk about the different forms of passive transport and how this exists in different parts of the body, and then we'll move on to active transport. This slide is just showing us the highlights of what we've already talked about with passive transport. Remember, big key things with passive transport is no energy is required. We do not need ATP for this to happen. What we're going to be doing is we're going to have these particles move from a higher concentration to a lower concentration. So therefore, it's going down the concentration gradient. And this happens naturally. So the particles are constantly going to move. If you take a look at this picture here on the bottom of the screen, you'll see we have about, 15 or so of these blue particles, or molecules, on the outside of the cell. So this is the outside of the cell. This is the inside of the cell, and you can see that it's separated by this phospholipid bilayer. So we have 15 on the outside, 15 on the inside, and then as time lapses with this x-axis down here, you can see that this high concentration starts moving down towards the lower concentration, and at the end of it, we have about a 50% sharing of these molecules. So we started from a high concentration, and those high molecules went to the lower until they were even. So why is this important? Why do we need to understand what passive transport is and how it works? Well, if you look at the bottom of the screen, you can see examples in the body maintaining blood pressure. Your blood is always going to go from high to low regarding pressure. So the pressure is highest in your arteries, and that's why when you take a blood pressure, the most accurate measurement is to squeeze off that brachial artery in your upper arm and basically occlude the blood near the elbow region, the antecubital region, but you can also take it at the wrist as well. You're cutting off artery flow temporarily. It's always going to go high to low, so when it comes back to the heart through the veins, it's really, really low, almost non-existent, and then the heart kind of resets that whole stage for us. So high to low, high to low. Same thing with kidneys. We'll have a whole chapter on the urinary system next semester, but your kidneys use passive transport to help filter things. And delivery of substances to the body, oxygen and carbon dioxide, a very just easy example to talk about regarding passive transport. Now that we've discussed what passive transport is, let's talk about the different types of passive transport. So we're going to introduce 4 different types. The first one being diffusion, and sometimes we call this simple diffusion. And what diffusion is basically what we saw with that first example that I showed you with the passive transport, with the little container or beaker, and it had a median, and the particles were separated with the concentration gradient. As soon as I removed the barrier, everything just kind of scattered and moved high to low to equal out and make a 50-50 range throughout the beaker. It was the same thing that we saw with the time lapse in the other slide. So diffusion is scattering, spreading of molecules until equilibrium is met. Now, these molecules are going to continue to move. They don't just stop once it's 50-50, but they're not going to go back to their crazy concentration gradient unless something forces them to do that. So, kind of like our phospholipid bilayer, our fluid mosaic model, stuff is constantly changing and moving, but for the most part, we're going to maintain this 50-50 homeostatic or equilibrium. Now, with diffusion, sometimes, well, really all the time, well, let's keep it simple. With diffusion, things really they don't have a lot of barriers. They kind of just go as they please, or wherever they're needed, high concentration to low concentration. A perfect example of that you can see on the bottom of the slide with oxygen and carbon dioxide. So these molecules are so small that they actually do pass through that phospholipid bilayer. Now, if they're a larger molecule, sometimes they'll have a little more difficulty, but oxygen, carbon dioxide, thankfully, can just go in and out of our cells and tissues and bloodstream just due to simple diffusion. So if you have a lot of oxygen in the bloodstream and not a lot of oxygen outside the bloodstream, oxygen can leak out if it's a really small blood vessel. And we'll talk more about that down the road. Same goes for carbon dioxide. With the examples that I've drawn so far, in the median of those beakers or containers that I drew, I kind of drew this hard wall, this hard block where nothing can get in and nothing can get out, and that's kind of unrealistic. For the most part, throughout our cell membranes, most membranes are what we call selectively permeable, and what that means is some things can get in and some things cannot get in, and some things can leak out and some things can't leak out, and it's based on, 1, the cell's needs, and 2, another thing is how big is this molecule. On the previous slide, I introduced that oxygen and carbon dioxide are just really small, so they can just kind of sneak through wherever they're needed. The larger a molecule is, the harder and tougher time they're going to have getting in and out of cells. So we may need some help sometimes with those. If you take a look at the picture on the right, you can see a very similar drawing to what we've talked about so far. You see a beaker or container, it's filled with water, and then we have some kind of solute or particle floating in the water. The left side, let's look at this picture first. On the left side of this first picture, we have a very high concentration of whatever solute, let's just say sugar. And on the right side, not that many. And then, but if you notice, take a look at the barrier or the membrane separating the two, and what you'll see is there's tiny little holes separating parts of the barrier. So these little sugar molecules are allowed to leak out or move from A to B, left to right, and over time, as you'll see, eventually, once again, for the most part, our body likes to be 50-50 or in an equilibrium-like state. Now we'll talk about exceptions to that rule in just a bit, but for the most part, diffusion, we're always going to try to equalize everything. Unfortunately, some of you may be familiar with dialysis, whether it's you or a loved one or a friend. Dialysis is a really great example of how this semi-permeable membrane idea actually works. With somebody going through dialysis, what's happening is their kidneys aren't working properly. And one of the kidney's main jobs is to filter our blood. It basically cleans our blood, and it's really 99% of our blood. And only 1% of that blood and that stuff floating in the blood actually gets turned into urine. So yes, our kidneys make urine, but even more importantly, we're cleaning blood. So if somebody's filtering system, their kidneys aren't working right, well, that's very, very dangerous. Toxins can build up very, very quickly and can be deadly if not corrected. So dialysis is a way of artificially cleaning the blood and pumping the clean blood back to the person's body. So how dialysis works is basically we take the blood out of the patient and we send it through a semi-permeable membrane. And we just get all the bad stuff out and put the good stuff back in. And depending on what type of dialysis, there's different types of dialysis, but some folks need to do this multiple times per week. Some folks, it just depends on how far gone your kidneys are too. But once again, particles love to flow from a high concentration to a low concentration. So as this blood is being spun through this apparatus, if we have a lot of toxins built up in the bloodstream, they're going to want to move or spread away or apart from each other. So they can move through a semi-permeable membrane, and then we can catch the stuff that's left over, the good stuff, and pump it back in. And then we can do that over and over and over again. Anybody who's been to a dialysis clinic, you know that it takes hours. It's not a 5-minute appointment. So by sitting there hours and hours and hours, you're cleaning the blood over and over and over again. We've talked about simple diffusion. Another type of passive transport is called facilitated diffusion. And facilitated diffusion is the same exact thing. What we're doing is high concentration to a low concentration. So if I have, let's say, 50 salt molecules on the outside of the cell, here's my plasma membrane, my wall, and I have 5 on the inside, generally speaking, those 50 are going to want to go towards the 5. We're still going to go high to low. The big difference with facilitated diffusion, the key word being facilitated, is we do need a little help. Sometimes these solutes or these particles are charged. They have a positive or negative charge to them. Sometimes they're just too big. Sometimes they don't get along with fat very well. So what we'll do is we'll have to sneak them through with the help of those proteins that we mentioned. So we don't require energy for this. Please make note, this is still passive transport. It's still going from a high concentration, that 50, to a lower concentration, that 5. But we are just getting a little help with the protein. The protein's not using any energy to do so. It's just kind of a road, like a detour route. And you can drive on the road and over and over and over again, the road's okay for the most part, right? You don't need to constantly repave the road every time one car drives over it. So facilitated diffusion with the help of proteins. Another type of passive transport is known as filtration. And inside of tubes throughout our body, in this picture you can see there it's a capillary, it's a small blood vessel, but inside tubes we have fluids, and those fluids exert a pressure, or they have a pressure against the inside lining of the tube. So picture your hose, your garden hose to water your plants, and if a certain amount of water is flowing through, we have pressure, and if you turn the nozzle more on, or a higher pressure, then more water would come through and exert a higher pressure. It'll start expanding and it will come out faster. So we have the same principle in our body where fluids push against surfaces and create this pressure. And depending on the pressure gradient, if the pressure is higher in one area compared to lower in another area, fluid will actually move out or inward, depending on which one's higher and which one's lower. So a great example of this, you coffee drinkers, is when you make a pot of coffee and you have a coffee filter, there's a pressure of fluid against that filter, and what happens is through tiny micropores of that filter, the water can actually drain and sneak through, right, due to the pressure. And then as soon as that pressure is no longer greater than it is on the inside of that filter, that top side of the filter, it stops, or basically until all the water's gone, right? So that is filtration. Remember, this is still a passive transport, so there is no energy required here. The pressure happens naturally. And then the last type of passive transport we're going to discuss is osmosis. Now, osmosis is similar to diffusion, but what we're using, it's mainly water through a selectively permeable membrane to balance things out. So I'll try to be careful here not to confuse you with the terminology, because a lot of folks just like associating osmosis with water, boom, done, next topic. But that's not really the case. What happens is, in some situations, solutes or particles are too big to the point where they can't pass through that semi-permeable membrane. So if you take a look at the picture on the right, you have two beakers, and let's focus first on the beaker on the left. And what you see is a semi-permeable membrane in the middle, and we definitely have a concentration gradient. Would you agree? We have about 7 particles on the left, and I don't know, let's say about 25 or so, 20 particles on the right, just eyeballing. So there is a difference, there is a concentration gradient, and what these particles would like to do is go high to low, right, to equal out. Now, what if these particles are too big? Or what if we don't have proteins to help them through? That concentration gradient is not going to want to stay there. They're going to want to balance, these particles are going to want to have balance, equilibrium. So what can we move? If we can't move the particles, maybe we can move something else to help try to balance this out. And as you can see on the picture on the right, what we can move is water. Water is a very, very small molecule, right? The molecular formula is H2O, so it's only 3 atoms. And by moving water, we can not exactly even things out in regards to the particle, but how much particle is floating in how much water we can balance out. So if you look on the beaker on the right, take a look at what happened. The particles are still the same. It's still 7 particles on the left and let's say about 20 on the right. But look what we did with the water. There's a lot more water on the right side now of the beaker compared to the left side. So what we did was we balanced out how much solutes or particles are floating in X amount of water. So picture making lemonade, right? You can make really, really good lemonade. You can make really, really bad lemonade. If you have too much sugar in the lemonade, it's going to taste like sugar. It's going to be too sweet. It's not going to taste well, or taste good, excuse me. So what can you do to fix that? You don't want to pour the whole thing out and start over, right? That's a waste of water and sugar and lemons. Just add water, right? Dilute some of that because you have too much sugar. And that's what we're looking at over here on this right side. We have way too much sugar, so when we drink that lemonade, it doesn't taste good. So all we're going to do is add water, and we're going to kind of dilute or spread apart some of those sugar molecules and make it taste better. What about over here on the left side? Let me clear some of this out. On the left side of our lemonade example that I'm running with, we don't have enough sugar in it. It just tastes like water with a very, very tiny hint of lemon. It's not that refreshing. It's not that good. I might as well just drink water. How can I make it sweeter? Well, let's add some sugar. Or What we could do if we don't have sugar is you can just pour out a little bit of the water. And that's kind of what we did here, where we're using the water from the diluted area and we're putting it towards the concentrated area. So yes, even though we have the same amount of sugar, we have less water now. So it should taste more like our desirable lemonade. So that's what osmosis is. It's a movement of water through a membrane to help balance out solutes that can't balance each other out because they're more than likely too big. And what causes this movement is similar to filtration. It's pressure. It's pressure of that water wanting to move from that higher concentration to that lower concentration. In this case, it seems that it's kind of moving in an opposite direction to help balance out those solute concentrations. So how water accomplishes this? Remember, water can't just move through the phospholipid bilayer. It doesn't work like that because what is the phospholipid bilayer made up of? Yeah, lipids, fats. So water and fats aren't going to mix. That's not going to work well. So we need a protein to sneak it through. And the protein needs to be very friendly to water. It needs to be hydrophilic. And we do have proteins all over our body that we can generate, that we can make, and that we can use and shut down and create more very quickly, especially in the kidneys. And these proteins are called aquaporins. And that kind of makes sense. Aqua means water, and porin is a pore. It's like an opening. And water can sneak through these aquaporins and do their job. And that is called osmosis. One more reminder, I know I'm a broken record, but I want to make sure you're understanding big picture. This is still passive. Water can move by itself without any extra energy needed. And we're going to use examples of salt and getting dehydrated or overhydrated on the next several slides to kind of bring that point home. Now that we've covered osmosis, we can introduce the topic of tonicity. And tonicity is just referring to the ability of water to move in and out of cells. So remember, the inside of your cells are mainly made up of water. The outside of your cells, the interstitial or extracellular space is what we call it, also mainly made up of water. So depending on our concentration gradients, whether it's salts or sugars or other particles, whatever, water is constantly moving in and out of the cells. Now, that's okay as long as we have the same amount of water going in as the amount of water going out. As long as that's somewhat equal, no big deal. We're going to always have around that same amount of water inside the cells, and we're going to maintain our shape. And if we maintain our shape, we maintain our function. The problem is if we have too much water leaving the cell or too much water coming into the cell, then we can start getting some pathology and issues going on. So what we're looking at here are two solutions. So we have water floating inside of a cell, and then we have water on the outside of a cell. And if they have a different concentration from each other, and they're separated by a semi-permeable membrane, which, as we said, most are, water is going to move back and forth. Now, just like simple diffusion, just like facilitated diffusion, just like osmosis, eventually what we're going to want is to reach some sort of equilibrium. That's the ideal, that's the goal. But as I mentioned, sometimes this gets a little wacky and this can cause some issues. We'll cover the normal versus the abnormal on the next slide. Here are the three different states that our cells can exist in regarding tonicity. I'm going to draw a line between isotonic and the other two, showing you that isotonic is our goal. That's our ideal that I mentioned on the previous slide. This is our norm. And the other two, this is more when stuff starts moving around. And I'm going to put abnormal, but that doesn't mean it's deadly or anything like that. It just means that you were possibly exercising or dehydrated or losing water in some way or fashion. If it gets too serious, it can be deadly. But for the most part, our body's trying to strive towards this isotonicity. So let's look at the definition and break down these words, these terms here. Isotonic. So two solutions. When we have two solutions, what we're referring to is the solution, the water inside of the cell, and the second solution is the water outside the cell. And then once again, inside of this water, we have all sorts of different electrolytes floating around in it. We have salts floating around in it, which I guess are electrolytes, and we have sugars and hormones and all sorts of different chemicals. And isotonic means, iso refers to same. So whatever is floating around the outside of the cell compared to the inside of the cell is pretty much the same, so water really isn't moving. There's not a lot of osmosis happening. And if there is osmosis happening, it's very, very minimal. We're pretty much at 50-50 equal. And once again, that's where our cells like to be at. So for any of you who wear contacts, perfect example, when you clean your contacts and when you preserve your contacts and when you put your contacts in your eyeball, you don't use water, right? Because water isn't equal to the fluid actually inside of our cells. It's not pure water. Like I said, it's water with electrolytes, salts, sugars, all those things in there. So what we use is we use a saline solution. And that saline has a mixture of the salts and sugars and everything that's very identical, or at least very close, to what actually exists inside of the human body cells. So can you squirt direct water in your eye? Yes, absolutely. But to truthfully clean the contact lens, what you really want to do is you want to have a saline solution. You want to have the same inside your cells as outside your cells. So there's not going to be a lot of water movement. Let's look at these two abnormal conditions. Hypertonic and hypotonic. We know these prefixes. We've seen these before. Hyper means above or greater than or increased. So if I have hyperglycemia, I have high blood sugar, right? And we've seen hypo before. We learned about the hypogastric region, the hypochondriac region. Hypotension means low or below or less than, decreased. Now, very important, in order to get these not jumbled in your brain, what you want to know is you want to know the reference point. What are we comparing to? Are we comparing the outside to the inside of the cell? Are we comparing the inside to the outside? What are we doing? And your answer is we are comparing the outside of the cell. So I'm going to put a 1 here on the outside to the inside of the cell. And that's what's going to give you an idea of what these terms mean. So let's look at hypertonic first. Hypertonic. What hypertonic states, or what a hypertonic solution is, the definition, it states a greater concentration of solutes than another solution. So we're comparing outside to inside. So all that means is this. We have, let's put some math to it. Let's say we have 300 solutes, and a solute is, once again, just a particle floating in water. It's just a molecule. We have 300 molecules outside the cell, and let's say we have 100 inside the cell. So we have a concentration gradient. These numbers are not equal by any means. Now, these molecules can't always cross. They can't always get through that semi-permeable membrane. So if we can't move the 300 to the 100 to make it 50-50, what can we move? What did we just talk about regarding osmosis? Exactly, water. We can move water from the inside of the cell to the outside of the cell to help dilute or balance or spread out that higher concentration. So a hypertonic solution means there's more stuff on the outside of the cell compared to the stuff on the inside of the cell. So know your reference point.
The reference point is it's the outside compared to the inside. And if the outside is higher, we use the term hypertonic. The cell is sitting in a hypertonic solution. Now let's flip the script. What if we have this? What if we have inside the cell, we have 400 solutes or particles, and we have 70 on the outside of the cell? So once again, concentration gradient is evident. And if we can't move the solute, we're going to move water. But what's going to happen is water is going to move from the outside to the inside. So this is what happens in a severe hypotonic solution. So how are we getting the term hypo? Remember, we're comparing the outside to the inside. So we're comparing this 70 to the 400. Is 70 smaller than 400 or is it greater than 400? And your answer should be smaller than. So since it's smaller than, our prefix, our root term is hypo. So if the outside is smaller than the inside, when talking about particles and solutes, where the cell is said to be sitting in a hypotonic solution. To fix this, osmosis is going to occur and water is going to rush into the cell. Now, once again, these are okay. This does happen every single time you don't drink water on a regular basis, or every time you work out, you do mess with these solutions, these concentrations. But your body has built-in mechanisms to help balance this out long term. Now, there are situations where this can be really, really severe, and we'll talk about that on the next slide. With this slide, we're going to talk about the extreme situations, what can happen if too much water is moving in or out of the cell. So once again, you can see on the right side with the picture, you can see our two extremes. We have hypertonic, where we have more solutes on the outside of the cell compared to the inside of the cell. And then we have our hypotonic, where we have less material outside the cell compared to the inside of the cell. And then we have our goal, our natural or ideal isotonic. So what we're looking at in this picture here are red blood cells. And if you look at isotonic, where I just drew the star, this is what your red blood cells should look like. They should look like these little, what they call biconcave discs. So they're kind of like these inner tubes, but they indent on the inside on both sides. So they're kind of like these little frisbee donut things, but there's no hole in the donut. There's this kind of like a sheet connecting all the parts to it. So that's a healthy red blood cell. If you look to the left and right of those pictures, you'll see that those don't exactly look the same. One, they're all kind of shriveled up and smaller, and the other one, they're kind of bloated and they lose that frisbee-looking shape. So let's talk about these two extremes. First, let's discuss hypertonic. In a hypertonic solution, what's going to happen is there's a lot of solute on the outside, so water is going to leave the cell to help minimize that effect or that imbalance. So if too much water too quickly leaves, what happens is the cell can actually lose its shape. And what we call that is crenation. And that's another way of saying shriveling up. That's another way of saying shrinking. But the proper term is crenation. And the crenation really only happens in a severe hypertonic solution. And once again, if we lose our shape, we lose our function, that red blood cell is not going to work properly. And that's not good. If you have too many red blood cells not working properly, we have a serious, serious issue. So to give you an example of this, this is why humans, we really can't drink the ocean water, right? This is why if you're going to get stranded in a body of water, you don't want to get stranded out in the ocean. You want to get stranded, hopefully, a lake or a river would be a little easier to survive versus an ocean. With the ocean water, it's so salty that it's going to create a hypertonic environment. We're going to have so much salt on the outside of our cells, water is going to leave our cells and that's going to cause them to basically shrink, shrivel up, and this word crenate. In a hypotonic solution, let's flip the script here. In a hypotonic solution, there are a lot of solutes on the inside of the cell compared to the outside. So what's going to happen is water is actually going to rush into the cell through osmosis. And if we have too much water rushing in, what can happen is we can also lose our shape. The cell will actually get bloated, and if it gets too filled up with water too quickly, it'll actually pop or burst like a water balloon, right? If you're filling up a water balloon with water, you can only go so far, so big, it will eventually pop on you. And what this term is called, this popping, is, well, it's hemolysis in the blood cells, but generally it's just lysis. If the cell bursts, that means the cell lysed or went through a process called lysis. But hemo means blood, so hemolysis or hemolysis. An example of when this can happen is pure water. Pure water that doesn't have any minerals or any extra electrolytes or solutes added to it is not isotonic to the body. Remember, our body does have a certain amount of salts existing on the inside of our cells and the outside of our cells and electrolytes. So if you were to chug pure water very, very quickly, and I'm talking really drastic here. And I heard this there was a radio contest several years ago, really unfortunate situation. There was some kind of dare, some kind of challenge to drink a gallon of water in under 5 minutes or 2 gallons of water in like a really short amount of time. And, it's funny and, yeah, let's see how much water we can chug. But the problem is if you drink water too quickly, your urinary system can't handle all that water coming in at once, and you put your cells in a hypotonic state. So water starts rushing into the cells because it has nowhere else to go, and your red blood cells start popping and bursting. And then what happens is you can actually run out of oxygen and you can die. And there was actually a story in California, unfortunately, where a person did die from this. It's called water poisoning. So even though water is probably the best thing for your body, too much of it in a very short amount of time, like chugging a gallon of water very quickly, is actually poisonous. It can actually cause your cells to pop. So these are the two extremes. Crenation, shriveling, is involved with a hypertonic solution, and hemolysis or just lysis is involved with an extreme hypotonic solution. But the good news is our body is really, really good as long as we're not in those extremes, as long as we're not too dehydrated, we're not exercising for several hours in 105-degree heat, or we're not chugging gallons of water without anything else in it. Just, throw some salt in there, throw some electrolytes, some pickle juice in there. Our body's pretty good at trying to maintain this isotonicity, this balance. Okay, so that is all passive transport, what we've covered so far. All of that happens naturally, whether it's simple diffusion, facilitated diffusion, filtration, or osmosis. There is no energy required for that. Now what we're going to do is we're going to flip the script and we're going to talk about active transport. And remember, the big thing with active transport is we're going up the concentration gradient, or we're going against the concentration gradient. So just to kind of give you an idea of that drawing, say that this membrane that I'm currently drawing on the screen is semi-permeable. We're going to have 1, 2 solutes on the left and let's say 8 on the right. What we're going to do in active transport is we're going to take these 2 solutes and pump them to the higher concentration. So that's going to go against everything we've talked about so far. So we're going to need energy in order to accomplish this. And what is the name of our famous absolute necessary energy molecule? You got it. You'll probably hear me say this 3-letter abbreviation more than anything this whole semester. ATP, ATP, ATP. This is your energy currency. We need this in order for active transport to be accomplished. So many different types of active transport. We're only going to cover 3 big types. We're going to talk about primary active transport on the next slide. And then we have 2 ways of getting things in and out of the cell, endo and exocytosis after that. So let's dive in. Before we start diving into the details of the primary active transport and the sodium-potassium pump, I kind of need to set the scene for us so we understand what's going on. Inside of your cells, we have a boatload of potassium. So to make numbers nice and neat for us, I'm going to use 100. And the abbreviation for potassium is K+. Remember, the plus is indicative of it being an electrolyte. As it dissolves in water, it separates and has this positive charge to it. Outside of the cell is not that much potassium. So to kind of give you an idea of how much, it's about 10 potassiums. So what you are seeing is inside the cell, we have 10 times as much potassium compared to outside the cell. Huge concentration gradient. Now let's talk about sodium. Sodium, it's flip-flopped. We have a whole lot of sodium outside the cell. And we do not have that much sodium inside the cell. And the ratios are actually pretty similar. So the abbreviation for sodium is Na. Okay. Based on what we've learned so far with simple diffusion, we know that these things want to mix and become more in an equilibrium-like state, right? That 100 potassium on the inside wants to move out of the cell and get more close to 50-50. The 100 sodiums inside the cell want to move, the 100 sodiums outside the cell want to move into the cell to make it more like 50-50. And guess what? That does naturally happen. That's what we talked about. That's what passive transport is. But in certain situations, we don't want this to happen. So let's talk about structures in our body that run on electricity, such as your muscles. Example, skeletal muscles and your heart muscle. Also, all of the nerves and other nerve-like things in your nervous system run on electricity. So guess what happens when those ions to the right, that 110, mix together and they become homeostatic, or in an equilibrium-like state, they become even. These muscles and nerves fire, they contract. So the nerves send messages, the muscles contract. Cool, awesome, that's great. Is that how the body works? Yeah, yes, it is. And that's great, that's good to know. Okay, so if I want my muscle to contract, I need potassium to, I guess, leave the cell, and I definitely need sodium to come into the cell. Cool, awesome, done. Okay, now that happens, but do you want your nerves working 24/7? Do you want your muscles firing 24/7, contracting 24/7? As you're sitting here watching this video, make a fist. Make a fist with both hands and flex your elbows, basically make a biceps. All right, and then bend your neck to the left or the right, and then curl your toes, bend your knees, flex your abs, and shrug your shoulders. And hold all those positions. Okay, we got a good another hour or so in this video, and I want you to hold it the rest of the time. And at the end of the chapter, tell me how you feel. Okay, I'm just kidding, but what I'm getting at is we don't want our muscles to contract all the time 24/7. How uncomfortable would that be if the muscle is in constant contraction, constant spasm? That'd be awful. Same thing with the nerves. You don't want your nerves firing all the time. Your nerves talk to your muscles, so if your nerves are firing all the time, your muscles can be hyperactive. Your nerves talk to your organs. We don't want our digestive system working 24/7. We want it to give it a little rest when we're not eating. We don't want our brain overworking. All of us may have experienced fatigue from time to time. We don't want to make that worse. So what we do is we use something called the sodium-potassium pump, and we go against the grain. We go against the concentration gradient to maintain this imbalance over here. So what we're going to do is this. There are going to be proteins all along the plasma membrane. If you can look, very small picture. If you can look, these proteins found in this phospholipid bilayer, there's proteins all throughout the membrane, and what they're going to be doing is they're going to be taking 3 sodiums inside the cell, and they're going to be kicking them out of the cell. And take a look at what we're doing. We're taking 10 sodiums compared to the 100 sodiums, and we're going to remove 3 of those 10. So we're going completely against the grain. Meanwhile, what we're going to be doing is we're going to be taking 2 potassium in to the cell. So we're going to take this low number and go low to high, which once again is a little weird, right? But what we're trying to do is avoid this 24/7 muscle contraction, this 24/7 overactivity of the nerves, you be in constant pain. So the sodium-potassium pump combats that. Now, because we're going against the concentration gradient, we have to use energy. Once again, our energy molecule, ATP. So we're going from low to high, which is complete opposite of what we've talked about with passive transport. And what we're doing is we're keeping the sodium in the cell very low. We're keeping that 10. We're actually making it less than 10 in relation to the ratio I gave you. And we're getting the potassium higher. So that way, even though some sodiums are naturally leaking into the cell and some potassiums can be naturally leaking out of the cell, we're maintaining this differential or this concentration gradient. Now, what we're going to do in the muscle physiology chapter and really the nervous physiology chapter is we're actually going to use numbers. We're actually going to put a voltage to this. And we're going to say that the very inside part of the cell membrane is actually negative compared to the outside part of the cell membrane. And that's what's going to keep us at rest. But for now, let's keep it nice and simple. We don't want 50-50 in our muscles and nerves unless we're using them. If my brain wants to use my fingers to type something or text something, if my brain wants to move my feet and my knees and my hips to walk, yes, then what we do is we shut this whole process down. We shut the sodium-potassium pump down and we let the potassium go crazy, flood out of the cell, and we let the sodium go crazy and flood into the cell. And that's when your muscles and nerves are working. But as soon as we want to stop walking, as soon as I want to stop moving my fingers, these sodium-potassium pumps get kicked into gear with the help of ATP, and we establish this concentration gradient again. We make all the potassiums go inside the cell, all the sodiums go outside the cell, and that's what lets us be at rest. Your muscles, your nerves are in chill mode. So, to summarize all this information that we just talked about, this is an active transport. What that means is we're going against the grain, and because we're going against the grain, we need ATP to accomplish this. What we're doing from a cellular standpoint is we are kicking 3 sodiums out of the cell. Meanwhile, we're bringing in 2 potassiums. Now, why would we not bring 3 sodium out or three potassium in? There's other stuff going on. Potassium kind of is really stubborn, and they're leaking out on the other side of the cell. So to maintain this concentration gradient, the magic ratio is 3:2. And remember, we have hundreds of these proteins all throughout the cell membrane. So that is primary active transport. We're going to be using a pump mechanism where we're pumping 3 sodium out, 2 potassium in. And the main pump, the only one that we're talking about here, is this. It's called the sodium potassium pump. Sodium out, potassium in. An additional form of active transport is endocytosis. Anytime you see the prefix endo, that always means inside or within. You're going to see that a lot this semester. Inside or within something. And cytosis means transport. We're moving something in the cell, out of the cell, within the cell. It's just movement. So we're, in this case, endo meaning in, cytosis meaning movement. We're moving stuff into the cell. Now, how this works. One, we have to have those receptors. Remember, those proteins have several different purposes. One of them, they work kind of like those satellite dishes, and they can communicate with other parts of the body. Now, once those receptors are activated, there are 2 ways of getting things into the cell. And it really just depends on whether it's a big thing or a small thing. If it's a big thing, we're going to kind of eat the structure, we're going to like swallow it. And what that's called is phagocytosis. And once again, cytosis means movement in the cell. Phago means to eat. So literally it translates, as you can see, cell eating. And that's when we have a very large substance, such as a bacteria or bacterium. And we literally just kind of swallow it. In smaller particles, such as liquid, we kind of just slurp it into the cell or drink it. And what that's called is pinocytosis. And that's referring to cell drinking. Now, if you take a look at this picture here, you can see that whether it's phagocytosis, pinocytosis, or we're dealing with a more specific type called receptor-mediated endocytosis, which is kind of a little more complicated, but what we're going to be really focusing on are the phago and pinocytosis. They're all doing the same thing. All 3 of them are doing the same thing. Whether it's a bacterium, we're going to kind of change the plasma membrane and kind of engulf it or swallow it. And then what we have is this phospholipid bilayer bubble surrounding the bacterium. Same thing in pinocytosis. In the middle, if we're drinking something, the phospholipid bilayer kind of indents, or what we call invaginates, and then it pinches off, and we get another phospholipid bilayer protecting and surrounding whatever we just drank in. And then same thing with the receptor-mediated example of endocytosis. We have a phospholipid bilayer surrounding those structures. So endocytosis, we're bringing something into the cell. What happens is the cell membrane kind of bends and folds and then pinches off from itself. Now that might sound like a bad idea, right? Phospholipid bilayer, we kind of need that to protect our cells. Well, guess what? We're going to have an organelle, which is a mini organ inside the cell that specializes in making these things. So as soon as we use them, we can just make more of them. No big deal. The last form of active transport that we're going to cover is exocytosis. Now remember, this is active transport, so the slide we just covered with endocytosis, that is active, so it does require energy. It does require energy to make those phospholipid bilayers. We do have to spend money in order to bring things into the cell. Same thing here. Same exact thing here. We need to spend energy in order to spit things out of the cell to create these bubbles of phospholipid bilayers. So you can see exo, if endo means inside or within, exo means outside. And what we're doing here is we already have a bubble made. And generally these bubbles are called vesicles. And this can be anything. This can be a packaged protein, this can be a hormone, this can be anything. And what we're going to do is we are going to fuse that vesicle, that bubble, with our plasma membrane towards the outer part of the cell. And then eventually, it will just spit its contents out into the extracellular fluid, or the outside space of the cell. And then that bubble will actually become part of the plasma membrane. Once again, though, this stuff isn't free. We do have to pay for it. So it does come at a cost, and that cost is we have to spend ATP in order to do this. So in recap, we've talked about passive transport and active transport. The main types of passive transport that we discussed were simple diffusion, facilitated diffusion, filtration, and osmosis. All of those happen passively, therefore no energy is needed. And active transport, we talked about 3 specific examples. We talked about primary active transport, and the big example was the sodium-potassium pump. And then we have this moving stuff in and moving stuff out of the cell. A lot of times this is called vesicle-related movement because of those bubbles. And those were endocytosis and exocytosis. So what I would encourage you to do is rewind this video 20 seconds and listen to what I just said. Make yourself a little chart, make yourself a little table. Make sure you know the differences between the 4 examples of passive transport that we talked about and the 3 examples of active transport that we talked about. So that way you can separate them, which one needs energy, which one doesn't need energy. And then make sure you know the specific differences between all of them. Now that we've covered everything we want you to know with the plasma membrane, we are ready to work inside of the cell. So remember, all cells in the body, at least 99% of them, have 3 components. Number 1 was the plasma membrane. That's what we just finished talking about. Number 2, we said earlier in the chapter, was the nucleus. I'll just put NUC for nucleus. And then number 3 is everything that's not the nucleus. So that's what we're going to be talking about next, is the cytoplasm and all the stuff floating in the cytoplasm, which we're going to call organelles. And organelles are just tiny little mini organs. And they all have their own unique shapes, and therefore they're all going to have their own unique functions. Remember that the cytoplasm is everything that is not the plasma membrane and the nucleus. So typically it is this kind of water jelly-like substance, and the majority of it is in fact water, and the fluid part, or the water part of the cytoplasm, is called cytosol. So what the cytoplasm consists of is the cytosol and all of the cell organelles, and all the proteins floating in the cytosol, which kind of makes it more of like a gel-like substance. So as I mentioned before, organelles are just these kind of mini organs. They're all going to be unique. We're going to kind of have a detoxifier organelle. We're going to have an energy-making organelle. We're going to have like a post office organelle that stamps things and sends it out and receives letters. We're going to have all sorts of cool things going on. And remember that some of them have membranes like the mitochondria and the endoplasmic reticulum, and some of them, like a ribosome, does not have a membrane. So for the next several slides, for the next like 15 slides or so, what we're going to do is we're going to talk about all the different organelles that exist in your cells. And we're going to basically talk about their function and why they're so important to our cells. First organelle we're going to discuss is the endoplasmic reticulum. So the endoplasmic reticulum is actually attached to the outside of the nucleus in a lot of cases, and there are 2 types of endoplasmic reticulum. On this slide we're only showing you one. You can see it's called the rough endoplasmic reticulum, also known as the RER. And then on the next slide, we're going to cover the other one, which is called the smooth endoplasmic reticulum, or the SER. So we have rough and smooth endoplasmic reticulum. And if you take a look at this first picture in the top left that I'm circling in red, you can see a subtle difference. The rough ER kind of has these red dots all over them, and the smooth ER is clean, does not have the red dots. So they are a little different in regards to what they look like, and therefore they're going to have a different function, right? Now you betcha. So those little dots on the rough endoplasmic reticulum, what they are ribosomes. And what we're going to learn in just a moment when we get to the ribosome page is this is where we make our proteins. So going way back to chapter 2, last chapter, I kind of spoiled it for you and I told you what we're going to be doing with our DNA and our RNA. So DNA is our genetic material. We're going to actually take our RNA and kind of use it as a map to eventually make a specific type of protein. So what we're going to do is we're going to send the RNA to these things called the ribosomes. And a big portion of the ribosomes found in your cell are actually located on the rough endoplasmic reticulum. So the reason why they're called rough endoplasmic reticulum is because all these ribosomes kind of make it look rough and grainy. If you were to picture like a magic school bus, or like if you were to shrink down to cellular size and take a tour of the cell, you would see that all these bumps make it look kind of rough and bumpy, where here in the smooth ER there are no bumps, and therefore we would call it the smooth ER. So what the rough ER does is indirectly it kind of helps with the making of the proteins, because the ribosomes are what makes the proteins, and the ribosomes are on the rough endoplasmic reticulum. But what the RER really does is it helps with that folding process. So remember, back to chapter 2, we had that primary, secondary, tertiary, quaternary of the folding of the amino acids. All the ribosomes do is they're just making them. They're just basically putting them in the correct order. They're just focused on that primary, putting things in the proper stage. Where that folding starts taking place is in the rough endoplasmic reticulum. Those alpha helices, those beta plated sheets we discussed, that's in the RER. And in a lot of cases, believe it or not, these proteins get messed up, they misfold, and we have to discard them. We have to throw them away. So the rough ER is kind of in charge of looking through all those things and sending the bad ones out into the cell so they can be destroyed. So we don't have messed up proteins going around our body, right? We don't want that. Another interesting thing that the rough endoplasmic reticulum does is it makes those integral and peripheral proteins. Well, we didn't really go into too much detail with that, did we? All those proteins that we talked about inside of the phospholipid bilayer itself, the things that serve as receptors or transport proteins, the rough ER is specifically in charge of regulating that as well. Now, at the end of the day, we're going to talk about the nucleus being the brain of the cell. Because the nucleus has the DNA, the nucleus is really in charge. But once the rough endoplasmic reticulum receives order or instructions, that's their job, is to help with this protein making, this protein synthesis. So let's move on to the smooth endoplasmic reticulum, and we'll learn that they do not help make proteins, they help make the other macromolecules. Alright, so with the smooth endoplasmic reticulum, also known as the smooth ER or SER, they once again don't have ribosomes. That's why they're called smooth. And they're going to be really focused on lipid synthesis. Now, you see in your notes that, yeah, the smooth endoplasmic reticulum makes carbs as well. Kinda, so does the rough. They both kind of make carbs. Those glycoproteins get put together with the rough ER. The big difference is, though, that the smooth ER helps make lipids. So, with all of that changing going on in the endo and exocytosis that we just mentioned with the phospholipid bilayer, if we are losing phospholipids due to too much endocytosis, no big deal. We can just make more as long as we have fat available to us from our diet, and the smooth endoplasmic reticulum is what helps us with that. So as you can see, it makes membranes for cells. It can also make membranes for the organelles, such as the mitochondria or
the Golgi apparatus or something like that. Another cool thing the smooth endoplasmic reticulum is responsible for is detox, specifically in the liver. Whenever we put a foreign drug or substance into our body, this type of organelle is what helps us break that stuff down. And these things can actually change in size and actually grow tolerant to certain drugs, and that's what happens in the example of like alcohol or medication. If you become tolerant to a drug where you have to take more of it, it's due to the smooth endoplasmic reticulum being so good at what they do. Your body's not exactly challenged in getting rid of that and detoxing that. But as we know, that's not a good idea, because your body will eventually break down if it's pushed too far. And then another cool thing that the smooth ER does, specifically in your muscles, is it stores calcium. We're going to talk about this more so in chapter 10 or 11, one of those. And that is going to be extremely, extremely important. Calcium, very, very, very vital. You can't live without calcium in your body. And just kind of hold on to that right now. We're not going to go into crazy detail or anything. Chapter 10, we're going to talk in great detail about how calcium is regulated and where it's stored in this smooth ER, specifically in your muscle. So, makes fats, which therefore makes the phospholipid bilayer. It can help with detox, and it stores calcium.
Moving right along, ribosomes are the next organelle, and real simply put, what ribosomes do, once again, they make proteins. They are the protein synthesizers. Now, they're kind of these they're made up of proteins. It's kind of funny. They're made up of RNA and protein that make proteins. And what they do is they are created actually inside of the nucleus. We'll talk about the specific structure that makes them in just a bit. And they're kind of released to go out on their own. And so they're all what we call free ribosomes, and a lot of them are just floating around in the cytosol, in the water of the cytoplasm. And then eventually some of them just kind of settle and anchor onto things, as we saw a lot of them anchored onto the rough endoplasmic reticulum. That's why we call it rough ER, because it's got all those ribosome bumps on it. And a lot of them attach to the outside of the nucleus as well. But regardless of where they're found, cytoplasm, ER, nucleus, whatever, their main job is to make proteins. So what we're going to do is we're going to send a lot of amino acids to the ribosomes, and they're going to take that RNA code, all those A, C, Gs, and Us, and they're going to line them all up in the proper order. So we're going to get that primary sequence before we start folding them of making an eventual protein. So end of the day, big picture, ribosomes make proteins. Our next organelle is the Golgi apparatus, sometimes also referred to as the Golgi body. And I like to refer to the Golgi apparatus as the post office of our cells. As you can see in the notes, the main function of the Golgi apparatus is to prepare those bubbles, those vesicle packages, and process them, and then eventually export them to various destinations, either inside the cell or out of the cell. But it also receives packages as well, coming from the endoplasmic reticulum. So think about what a post office does. A post office receives letters from you, right? If you want to mail something to, let's say, Los Angeles, you're going to go to your local post office or just dump it in the mail. Eventually, it will get to your local post office, and they're going to process it. They're going to stamp it and put a barcode on it, or put a label on it to where it digitally says, okay, this is going to have to get to Los Angeles by this date. So then as it's getting processed, eventually it's going to get shipped out to a bigger center, wherever that may be, and then eventually will get to Los Angeles. And then whenever it gets to the Los Angeles post office, that Los Angeles post office will process it, stamp it, make sure that it's arrived, and then it will get it to its actual specific destination, whatever address it is in Los Angeles. So that's what the Golgi apparatus does. It receives these packages, these vesicles, and remember, really anything can be in these vesicles, specifically lipids and proteins. But we can be receiving packages of all sorts of different shapes and sizes. And it processes them, and then we also are able to export them. So importing and exporting, coming and going of vesicles is what the Golgi apparatus does. It monitors them, modifies, packages them, and then kind of sends them on their way. The next organelle are the lysosomes, and the lysosomes are really cool. I like these. They are specialized vesicles that come from the Golgi apparatus, and their job, I refer to them as the garbage collectors of the cell. They like to take out the trash. So specifically, they break down things using a set of enzymes. And remember what an enzyme is. An enzyme is a protein that specializes in catalyzing a reaction or speeding up a reaction, making things a little more efficient. So if you look down here at the bottom, you see all the different functions. We are able to break down proteins that are unnecessary inside the cell. We're able to break down and destroy defective organelles. If we have some ribosomes that are supposed to be making X amount of proteins per day, and they're only making half of that, well, maybe it's defective. Maybe it's just old. Maybe we don't need it. So let's break it down. Let's recycle it. And then we can make some new ribosomes. So just efficiency. And then we can also break down harmful bacteria and other substances with our lysosomes. So really, really cool picture on the right. If you kind of just take a look at this, you have a cell, and there is a bacterium in red that is approaching the cell membrane. And through endocytosis, specifically phagocytosis, which means to eat, the cell actually is able to swallow the bacterium inside of this big bubble. And that bubble, as you can see in the picture, is called the phagosome. And then we have our garbage collectors. The lysosomes can actually interact with that phagosome and secrete its enzymes and actually digest and kill the bacteria. And as you can see down here, our end product is we just have all these little bits and pieces of the bacterium that's not going to be harmful to the cell because we killed it. And then we can just spit out all of its contents out of the cell via exocytosis. So lysosomes really, really good at taking out the trash, getting rid of things that are either harmful or we don't need inside of our cells. Our next organelle is the mitochondrion, or mitochondria, plural. And the mitochondria, you may have heard of this phrase before. This is the powerhouse of the cell. This is the energy factory, the energy makers. This is where we're going to be generating the majority of our ATP. Not 100%, but pretty close to it. So we're going to have multiple membranes, and we'll get more into that in detail next semester, but these multiple membranes help us with organizing certain enzymes to help us make a lot of ATP down the road with the help of sugar and oxygen as well. Now, as you can see, the number of mitochondria in a cell depend on how much work is needed by that cell. So, for example, your certain cells near the surface of your skin, they don't do too much, right? Your skin is there to protect you, yes, but there's not a lot of crazy things going on physiologically with your skin compared to your muscle cells. Think about all the energy that you need in your muscles. I mean, just think about the cheek and tongue and jaw muscles right now just to talk. Think about all the finger muscles that you use to text and type all day. So our muscles, especially in our heart as well, have a lot more mitochondria compared to other cells throughout our body. Another unique characteristic of mitochondria is they actually have their own set of DNA, their own set of genetic material, which is really interesting to think about. Believe it or not, most of this, depending on what research you read, most of this mitochondrial DNA comes exclusively from your mom. And your mom got it from her mom, so really it's only traced along the female ancestry. So even though I'm a male, all of my mitochondrial DNA, most of it, or 100% of it, depending on the research you read, is from my mom. And then if I were to have a sister, the sister would also have it from the same mom. So you can trace mitochondrial DNA through the grandma, great-grandma, great-great-grandmother. And this is just another way in regards to forensics. There was a really cool study with a Russian royal family that they found remains in an unmarked grave, and they were able to trace it through mitochondrial DNA to a recent relative that this was your great grandmother that was buried here in this grave. It's pretty neat. But main function of mitochondria, ATP production. ATP, this is where we're going to get our energy. Next we have the peroxisomes, and the peroxisomes, if you take a look at the picture on the right, is also just like the lysosome. It's a bubble. It's a specialized vesicle that has a different type of function. Their function is to detox and destroy harmful substances. And you can look at the second bullet point and see that a lot of these happen in the liver. So peroxisomes, along with your smooth endoplasmic reticulum, really helpful in detoxing drugs such as alcohol. And then your kidneys also process a whole lot of drugs as well. A lot of our pharmaceutical drugs have to be processed and go through the kidneys, right? And peroxisomes are very helpful in accomplishing that. And they do it similar to lysosomes. They accomplish this goal by the help of certain enzymes. Peroxidase and catalase are just a couple of them. And that's really it. They also help break down fatty acids, but detoxing chemicals is the big one. So just one more thing I want to point out to you. Notice that this structure, yes, it's a bubble, but notice what's around the bubble. It's a phospholipid bilayer. So just like our plasma membrane, any type of organelle that has a membrane also has a phospholipid bilayer, that those two layers of fatty acids, and the fatty acids are kind of oriented towards one another, so they create this barrier to separate water from the inside and water from the outside. Next we have the cytoskeleton, and the cytoskeleton is kind of what it sounds like. Cyto means cell, skeleton is the skeleton, so just like your bones make up the skeleton of your body, what we have are proteins that make up the skeleton of the cell. So your bones make your shape, right? They support your weight and they give you your shape. The reason why my arm is long and skinny is because the bones in my arm are long and skinny. So the cytoskeleton, these are really made up of proteins, and the proteins determine the shape of the cell. So remember that some cells are going to be flat, pancake-like, some are going to be square, some are going to be weird rectangle, some are going to be looking like frisbees, some are going to be spider alien-looking things. And it all just depends on what's inside of it, making up that shape. So we're going to talk about a few of these fibers down the road, specifically in the muscle, but for right now we just need to know that the cytoskeleton gives the supporting framework or the shape of the cell. The next two slides are going to cover microtubules in and on the outside part of the cell. What these are certain specific structures that are made up of proteins, and although they look very identical or very similar under the microscope, they are different, and therefore they do have different functions. So we have two on this page. First are the cilia, and what the cilia are, they literally look like these little nose hairs under the microscope, and they kind of work like nose hairs if you think about it. What is the function of a nose hair? There's nothing pretty, there's nothing attractive about having hair in your nose, right? But it does have a function. It is important, and it's to catch stuff, catch junk, right? All the pollutants that we constantly breathe in, the nose hairs catch it, and eventually will form kind of mucus or boogers, if you will. And that's what the cilia does in your body. So cilia are these kind of nose hair looking things. Once again, they are proteins though. They're not hairs. They just look like hairs. And they're found in various places in the body, mainly going to be found in the upper respiratory area. So kind of like back of your nose, upper throat area, going down towards your lung area. And these little hair-like things, what they do is they move back and forth in these like wave-like rhythms to filter stuff, to move stuff out of the way. So what they do specifically in our upper chest area is any type of mucus or bacteria or fungus or anything, any bug that gets caught in that upper chest area, they actually move that material up towards our throat and mouth area. And a lot of times this is what causes our cough reflex, is the tickling of these little thousands and thousands of cilia moving back and forth. You also have this in your brain area and spinal cord area. And what they do is they move the fluid. Same thing, all these hairs kind of work together and they move in one direction to move the fluid in one direction. And their whole purpose is just to make sure that the fluid is not going the wrong way. So cilia, very, very important. And you can take a look at the picture on the right, the first diagram. It's showing you a small child who actually is suffering from a congenital disorder called primary ciliary dyskinesia. And so what that means is dyskinesia means the inability to move or bad movement. So the cilia in this patient or this person does not move well. And like I said, this is a congenital condition, so she was unfortunately born this way. Nothing that she did to cause this. It's just due to faulty genes from mom and dad. Something got crisscrossed in the forming of her as an embryo or fetus. And what happens is her cilia doesn't work well, so all of that junk that constantly gets filtered every day for you and I, hopefully, doesn't get filtered for her. So you can take a look at the picture here on the bottom right, and what's happening is we're getting a lot of mucus and bacteria and buildup on top of the respiratory tissue. And what that causes is a lot of infections. And that can lead to pneumonia and more serious things, unfortunately. So cilia, very, very important to avoiding that stuff, avoiding bad things getting into our lungs, because really the only thing getting into our lungs should be air. Right? If you swallow water or food and it goes down the wrong pipe, right? The reason why we cough so violently is because the cilia are irritated and know that that stuff shouldn't be there. So they cause that cough reflex. Now, these cilia can be damaged and paralyzed with a lot of trauma. So, for example, people who smoke, whether it's cigarettes, marijuana, vaping, any type of irritant that is going through the respiratory system can eventually lead to cilia being destroyed, or if not totally destroyed, paralyzed. And if you've ever noticed somebody who smokes a lot, their cough is a little different, right? They have a lot more phlegm and a lot more junk inside of their lungs compared to somebody who may not smoke. Same thing, you have tiny little cilia in your ears that help you hear, believe it or not. They're called stereocilia. And there's a lot of research that shows that smoking and certain drugs lead to the harmful effects and paralysis of these cilia. So Google it. There's a lot of links that smoking leads to hearing issues and hearing loss. So you want to make sure that you're not irritating these very, very important proteins that help keep you healthy. Then after the cilia, we have the flagella, or flagellum is one. And flagella are these cool little tails at the end of these sperm cells in the picture. So I'm going to highlight them in black. So you see the little purple circles, those are kind of the heads. And then you can kind of make out the tail on these things. Here's a tiny one right here. There's a tail there. So this is only found in the human male. So certain protozoans and other weird organisms and species throughout our Earth have flagella. Females don't have flagella, or at least none of their cells do.
So this is only found in guy sperm, and they look like these little tails, and that's kind of what they are, and their whole function is to help swim. So the sperm's job is to swim through the male reproductive tract, leave the male, go through the female reproductive tract, and eventually swim towards the egg to fertilize and make a baby. So with this tail, we need a lot of energy. So what do you think is located all throughout the tail in sperm cells? What organelle? If you were to guess, Golgi apparatus, or mitochondria, or endoplasmic reticulum, or lysosomes, if we need a whole lot of energy, which one would be found in the flagella? And remember, our energy factory are the mitochondria. So there are boatloads of mitochondria found in the sperm cells, specifically in their flagella, help them swim a very, very long distance. Our last organelle that we're going to cover are the microvilli. And the microvilli, although they look similar to cilia, they are very, very different, and therefore they have a different function. The main job of the microvilli is to increase a cell's surface area. And what that means is we're trying to just make this really small cell a lot bigger. So picture with me, I'm going to draw this cell for us here. And this red line is going to make this kind of circle, oval looking cell. Okay, so say that that's a cell, right? If we were to stretch that cell out like a string, if we were to cut it and then stretch it out like a string, it would measure a certain distance, right? But what if we did this to the cell? What if I have that same outer plasma membrane, but then we have all these kind of hills and valleys and extensions up and down and up and down and up and down, and you kind of get the point here. What if I was to cut this line and stretch it out like a string? Which cell is going to be bigger, which cell is going to be longer? And your answer hopefully is the one we just drew, right? We have all of these kind of hills and valleys and crevices and peaks and mountains and all these things that make this little tiny microscopic cell actually huge, gigantic. And we're going to talk about this phenomenon with our DNA as well. The DNA, which is found inside your nucleus, really small and compact. You need very special microscopes to actually see the DNA. Well, actually, you can find it with a compound microscope sometimes too, but if we want to actually see great detail, you need some specialized microscopes. But if we were to stretch out the DNA of one cell, so just take a hair or take a part of your fingernail, and if we were to stretch out that DNA in a straight line, it will be 5 to 6 feet long of all those letters, those nucleotide bases, the A, C, T's, and G's. 6 feet long of those letters. And that is an absurd amount of genetic information. But the reason why we can fit it into this microscopic structure is because we take all those A, C, T's, and G's and we just kind of roll it up and fold it and condense it and twirl it and swirl it into this big blob. So we have this really long big structure, and it's condensed in this small area. And that's what the purpose of the microvilli are doing. The microvilli is making this really small cell, or sorry, really big cell, and it's helping it fit into a very small space with all of these hills and valleys, all these extensions. So what is the main purpose of this? Specifically, we have a lot of these in our small intestine. We also have a lot of these in our kidneys. The main job, all of that surface area helps us with absorption. So, if you picture your gut, if you picture your small intestine, let's draw two tubes. Here's tube number 1. Nice straight tube, right? Let's say that that's your small intestine, and we just ate some pizza, and here's your pizza molecules floating around. certain pizza molecules are actually going to leave the tissue and go into the bloodstream so we can get the nutrients to all other parts of our body. And then a lot of things are actually going to go to the large intestine, and we're eventually going to get rid of it through fecal matter, through waste. But let's draw tube number 2. What if tube number 2 looks like this? From a microscopic perspective. I'm trying to do this without a mouse. My fingers are getting very tired. There we go. Okay, so they're kind of stacked on top of one another, so you can make the argument, yeah, tube 2 is the same distance as tube 1. But we know that if we were to pull or elongate or stretch out tube number 2 is very significantly longer than tube number 1. So let's take that same pizza, all those pizza molecules, and now what happens is you can see we have all this extra space. This tube is a lot longer, even though it doesn't appear longer. A lot of these molecules will be able to escape the tube and be absorbed into the bloodstream, and very few will actually leave as fecal matter. So because we have all these folds and wrinkles and curves, we're able to maximize absorption, and that's the whole purpose of why we eat in the first place, right? Why do you eat food? It's not because it tastes good, it's because we need our nutrients to keep us going and keep our immune systems and our bodies healthy. So that is the purpose of microvilli. Makes these really large cells fit in a very small space to increase absorption. Okay, so now that we're done with the cytoplasm, make sure, I would encourage you to go back and review all the organelles and their functions. Look at Google image some of the different organelles. We did provide you with pictures with the mitochondria and the lysosomes and the Golgi apparatus and the endoplasmic reticulum. But the more pictures you see, especially if you're a visual learner, it can help you kind of cement what these things look like and their functions. The mitochondria always kind of look like these weird kidney bean looking things. The lysosomes, peroxisomes look like these bubble looking structures. Ribosomes look like just these little dots compared to the size of everything else. So go back, make yourself some type of chart, and make sure you understand the functions for each organelle. So we've covered the plasma membrane, we've covered the cytoplasm, so we've covered all parts of the cell except the nucleus, and that is our final topic for the chapter. A few important characteristics with the nucleus. Generally speaking, for most cells, the nucleus is going to be the largest structure. And the reason why it's so large is because that is where our genetic material is going to be housed. That is where your DNA is located. And we have a lot of DNA, as I mentioned previously. We're going to get a little more detail in just a sec. Another thing that's found inside the nucleus is something called a nucleolus. Now notice the spelling, notice the pronunciation, nucleus versus nucleolus. So they are different. This whole picture right here that I'm circling in red, that is the nucleus. And then that purple or darker purple sphere circle looking structure in the middle of that, that would be the nucleolus. So it's part of the nucleus. Now, what does the nucleolus do? What's its function? Well, it helps make ribosomes. So as ribosomes start to degrade or not work properly, we can always just make more. So remember that the ribosomes are what's responsible for making our proteins. These are the protein factories in the cell. That's what's going to line up the amino acids in that primary sequence. And then eventually we're going to start folding everything and getting it ready. So the nucleolus is what makes your ribosomes, which makes your proteins. So you got to have the nucleolus working in order to make proteins in your cells. Another important feature of the nucleus is similar to other membrane-bound organelles, we have a phospholipid bilayer surrounding it. And instead of saying phospholipid bilayer of the nucleus or cell membrane of the nucleus, there's a
distinct term. It's called the nuclear envelope. And as you can see, it's membranes with pores, so we can selectively choose what comes in and what comes out. So we're going to be sending things like RNA out of the nucleus, and we're going to be bringing in things such as hormones into the nucleus. Now, the nucleus is the brain of the cell, for the most part, because the DNA, the genetic materials inside the nucleus, we can choose what genes are on and off. Well, I shouldn't say we. The nucleus can choose which genes are on or which genes are off, and that can dictate what kind of RNA is made and what type of proteins we make, and the proteins go throughout the body and have their own unique different functions. So we are constantly losing cells. We are constantly killing cells. Your skin cells die every single day. Your stomach lining cells die every day. Your kidney cells, they die. Your liver cells, they die. A lot of things die. So we have to make new ones all the time. And how we're going to make new cells is from our original DNA molecule. So I'm sure some of you have heard of the company, or you've seen the commercials, 23andMe, right? So 23andMe is a genetic company. They can tell you where you came from and your ancestry and all that stuff. And there's other places like Ancestry.com and all those things. But what we have in every single cell in our body, minus our sex cells, is we have 23 pairs of chromosomes. So we have 23 from your mom and 23 from your dad. So total, what we have is we have 46 chromosomes in every single nucleus of every single cell in your body. So as we make new cells, what we need 46 chromosomes in every single cell, so they're identical. Right? Your eye color, for the most part, hasn't changed since you were 2 or 3 years old. You've always had the same eye color. Your hair color can change. Your hair is a little different. Skin color, for the most part, doesn't change over a lifetime. You are your skin color because that's what your mom and dad and your grandparents gave you. If you're 5'4", at this point that you're listening to this video, you're probably going to stay 5.4 or shrink from this point on. Your bones aren't going to get any longer. You're not going to be making new bone cells or new cartilage cells to elongate the bones. So all of our cells, for the most part, stay the same when we're talking about how many chromosomes are in the nucleus. So if I need to make new skin cells, if I need to make new bone cells or new liver cells or new small intestine cells, I need to make sure that I have 46 chromosomes. So what we're going to have to do is take one cell and make two. Now, if I split this cell into two pieces, what we're going to get is two unhealthy, unviable cells. Because what happens when you split 46 in half? You get 23 in one cell and 23 in another cell. And those are both, that's not going to work. Those are not human cells. Unless we're talking about sperm and egg. So, we have a dilemma. How are we going to make all these new skin cells, bone cells, nose cells, mouth cells, everything? And the answer is we have to copy everything. We have to replicate this. So what we're going to do is we're just basically going to copy and paste. We're going to take our entire genome, every single letter, A, T, T, C, C, A, G, G, T, and we're going to copy it. And it's going to be an identical copy. So for a time in that cell's lifetime, we're going to have two copies of 46 chromosomes. So actually a total of 92 chromosomes. 23 from your mom, 23 from your dad, and then we doubled it all. And then what happens if we split this nucleus in half? Well then, awesome, we can actually get 46 chromosomes in one cell, 46 chromosomes in another cell, and we just doubled the amount of cells we have. We started with one, now we have two.
And what happens if those two do the same process? We started with 2, now we have 4. Double that again, we have 8. Double that again, and you get where I'm going. So that's how we make new cells. We copy every single piece of your DNA. And that's why your skin doesn't really change. Yeah, you can go out in the sun and get darker, regardless of what ethnicity or wherever you come from. But after 4, 5, 6 weeks, your skin color will go back to the original color that your parents gave you, unless you continue to go out in the sun. So we're going to get into more detail with this in just a moment. But let's go on to the next slide and review the differences and similarities between DNA and RNA. So you can see in this slide the characteristics of RNA. Once again, we said that RNA is single-stranded because we're getting that from the DNA template. We're going to split DNA open, and instead of saying ACGT, we're going to say ACGU, as you can see down here. Different types of RNA. We have ribosomal RNA. What we're going to introduce in just a moment is messenger RNA, and then we'll have transfer RNA. It's just different types, different purposes, but we're all really working together to eventually make our end product. The whole purpose of RNA is to give those ribosomes the code or the instructions of the map to make proteins. So that's our whole goal here, why we make RNA. Or we want to make proteins. We want to build muscle, we want to repair bone tissue, we want to make this in our cells and that in our cells, we want new ligaments and cartilage and tendons. We need to repair all that stuff every day. So we need protein. When we look at DNA, remember DNA is not single-stranded, it's double-stranded. It kind of makes that alpha helix corkscrew looking appearance. Instead of ribose as a sugar, we have deoxyribose, so we're missing an oxygen. And the nitrogen bases are the exact same except for the U, except for uracil. So we have A, C, G, and T. That's the big difference, as we talked about in Chapter 2, Chemistry. And once again, DNA is what's going to allow us to make your RNA. And once we have the code for RNA, then we can make your proteins. But you can't make proteins without RNA. And you can't make RNA without DNA. So DNA is the foundation for this whole endeavor. So once again, as we make cells, there's billions and trillions of cells in your body, and we need to make new ones because we're constantly killing these things off. So how we make them is something called DNA replication, also known as DNA synthesizing, DNA copying. And basically what we're going to do is we're going to take all those 46 chromosomes and make 2 copies of them. So as you can see, one parent strand of DNA, a parent strand meaning the original DNA, is replicated into 2 daughter strands. And why we say daughter, I honestly don't know. It's just we have a parent cell and they make 2 daughter cells, or 2 strands, which are going to make cells. And what we're going to have eventually is that 92 chromosomes, 2 sets of 46. So as I mentioned in the previous slide, that the entire genome is copied. So if you look at this picture, really neat how this is, what's going on here is you can see this is a chromosome. So once again, you have 46 chromosomes in your non-sex cells, so all the cells in your skin, bones, muscles, everything that's not sperm and egg, you have 46 of these. 23 from mom, 23 from dad. And if you uncoil them and unravel them, as you can see in the picture, you get just millions and millions and millions of these letters, these nucleotide bases, or these nitrogen bases, all those A, T's, and C, G's that we learned about. So what we have to do is we have to unwind, in order to replicate, we have to unwind this DNA, and we have a special enzyme that helps us do this. Remember what an enzyme is, I'll point to it here. It's called DNA polymerase. But an enzyme is a protein that just speeds something up. So this enzyme, what it does is it helps speed up the splitting apart process. And what's going to happen is we're going to make two copies of these letters. So take a look at, well, let's look at right here. This will be easy. Where I'm circling in purple currently. You see that T is in yellow and A is in red. So A and T always match together, C and G, G and C, T and A. And then once we get down the line, what we're doing is we're unraveling them. So we're separating these two strands. And then what we're doing is we're making the exact identical copy. So at the end of this process, what we're going to have is we're going to have two alpha helices strands. Or two pairs of alpha helices strands. So we're doubling the amount of DNA inside of the nucleus. So this next slide covers the central dogma of biology, and we've been talking about this. This isn't new information, but I do want to introduce the concept of the central dogma, which is just a really, really important thing to know. You can see the definition of what this means. Central is the center, main idea, and dogma is the belief, principle, whatever we're talking about. So the central dogma of biology is, and biology is just the study of life, right? So whether we're studying humans, dogs, cats, kangaroos, whatever, we're studying how we live. And a big part of how we make proteins that run our entire body is through our DNA and RNA. So that's this central dogma. And we've covered this before, right? We make RNA from our template DNA, and then we use the RNA as the treasure map or the code to make these specific types of proteins all throughout your body. Whether it's a building block protein that goes in your muscles or your tendons or ligaments, or it's a hormone that tells your thyroid to do this. There are millions and millions of proteins in your body. So what we're going to introduce on the next few slides are the two processes that get us from DNA to RNA and from RNA to protein. And as you can see here, they are called, number one is called transcription, and number two is called translation. So let's talk about transcription first. Transcription is copying or transcribing something. So, for example, if I'm a medical transcriptionist, what I do for a living is I listen to a doctor's notes. Like, for example, if a doctor's talking into a cassette or like a recording device, I am going to listen to that recording device and type it all out. So what we're doing in transcription is we are taking our DNA message, or our DNA code, and making that RNA message. So, for example, if I had, and we'll show you a slide on the next slide or in the future, but if one strand of my DNA says ACG, and then we'll say TA, let's do that. For my RNA, what I'm going to do is I'm going to pair it with a U, and then that's a G, C, T is best friends with A is best friends with U in RNA, right? We covered that last chapter in the chemistry chapter. So that is what transcription is. We're taking our DNA and we're making an RNA molecule from that. And what we're going to call this specifically is messenger RNA in just a little bit. Okay, so that's happening in the nucleus because the nucleus is where your DNA is found. Once we make the RNA, we're going to spit it out of the nucleus, and it's going to go to the protein factories. And those protein factories, remember, they are called the ribosomes. And the ribosomes are everywhere in the cytoplasm. They're floating around in the water. They're on the outside of the nucleus. They're on the rough endoplasmic reticulum. They're everywhere. But remember, this is a code. And what we have to do is we have to decode the code, or we have to translate the code. So what we're doing is making proteins. Now, who can answer this for me? What are the building blocks of proteins? What is the monomer for a protein? And what you should hopefully be saying is an amino acid. Very good. 20 amino acids that we need in our body. All those amino acids in certain forms and everything make up your proteins. So, what we have to do once we get to the ribosome is we have to translate this message. This UGCAU is in Russian, and the ribosomes speak amino acid. Let's say something that starts with an A. Well, Argentina, but that's Spanish. All right, but Spanish works. Let's do that. Spanish. So the RNA molecule is in Russian, and the amino acids are in Spanish. So that doesn't mix, right? Right? Somebody who speaks Russian is not going to understand somebody who speaks Spanish, and vice versa. So what we have to do is we have to translate that message. We have to translate this Russian, the purple, the RNA, to the Spanish, to the amino acid. And I'm going to show you how we do that in just a little bit. So transcription is taking our DNA and making RNA, and then translation is we're translating that RNA code to make proteins with the ribosomes. So let's continue and see how this actually works.
With transcription, first things first, this only occurs in the nucleus. Why? Question is, what are we transcribing? Where are we starting this? What's our platform? What's our basis? What's our foundation? DNA. We can't do anything without our DNA. So the only place in the cell where DNA is found, our genetic material, not counting that weird exception with the mitochondria, whereas most of your genetic from your mom and your dad found is in your nucleus. So this is only happening in the nucleus. All right, so how do we do this? What are we doing? We're trying to make RNA. Specifically, it's called messenger RNA, little m RNA, because this is going to be our messenger that leaves the nucleus and it goes to the ribosome to deliver that code. So several steps, and you can see this on the picture on the next slide. But what's happening is first we have to unwind the DNA and unzip it. And I showed you there's an enzyme that helps us do that. It basically just temporarily splits apart the DNA, just like we did with DNA replication. We're going to split it apart and make a copy of something. And then instead of an enzyme called DNA polymerase, we have a different enzyme. It's simply called RNA polymerase. And what we're going to do is, instead of making DNA, we're going to make RNA. So instead of adding A, C, Gs, and Ts, all we're going to do is add A, C, Gs, and Us. That's it. So then once we make all the A, C, Gs, and Us, that enzyme falls off. The DNA goes back to the way it was, which is kind of cool. So we don't damage the DNA by doing that. All we're doing is basically, it's kind of like Velcro, if you will. Say that you have Velcro shoes or a Velcro pad or something. All you're doing is you're unlatching the Velcro, doing what you need to do, and then you just adjusting your foot with your Velcro shoes, and then you put the Velcro right back. Boom. No big deal, no damage, you're fine. So that's what we're doing with our DNA. So this is not a damaging process. And then what we're going to do is once we have our messenger RNA processed and ready to go, we are going to send it into the cytoplasm. Why? Why are we sending the RNA into the cytoplasm? Well, because our next step is to bring the code to the protein makers. And where are the protein makers found? They're found outside the nucleus, right? Anywhere that's not considered the nucleus is called the cytoplasm. So even if it's just on the outside of the nucleus, if it's the rough endoplasmic reticulum or it's just floating around in the water, that's all cytoplasm, where those ribosomes are housed. So let's go on to the next picture, or next slide, and show you what this looks like. So once again, we have our cool chromosome, and we're just unwinding it to the point where you can see our DNA. And this top line here that I'll highlight in red, or point to in red, that is our DNA. How do I know that? How do you know that? Well, look at the letters. You have an A and a T. If it's a T, it's got to be part of the DNA. And if you notice, this A lines up with this T. this T lines up with this A, this G lines up with this C. So we split this entire DNA molecule apart, and now we're transcribing or making our RNA molecule with the help of this enzyme. So you can see what we're doing here, and we'll practice this on the next slide because you will have to do this. A is always going to be best friends with U in RNA. T pairs with the A. G is best friends with C. A is best friends with U, and so on and so forth. So this is transcription. It's happening in the nucleus. So this is a great opportunity to help you practice. What you have in front of you is a DNA molecule. So it is double-stranded. We have T's connected to A's and G's connected to C's and vice versa. In order to make a messenger RNA copy, what we have to do is first split the DNA, as you can see. So now that the DNA is split, what we're going to do is we're going to take a parent strand and we're going to make a copy. So we need to figure out who is best friends with who. So let's take this bottom strand, and what we're going to do is A is always best friends with what? U. Exactly. And then you should be able to fill out the rest of this. G is always best friends with C. That doesn't change. And then C. T is always best friends with A. A is always best friends, once again, with U. Good. And then G, A, and you get the picture. And then what we're going to do, as you just saw that happen, we're going to send it off out of the nucleus into the cytoplasm where all those ribosomes are hanging out. So that is transcription. That's what we're doing. We split the DNA, we make an RNA copy, we send it out, and then that DNA is going to come right back together and continue to do its thing. When we move on to translation, now we are in the cytoplasm. We are outside of the nucleus, and we're going to have three big players involved with this. We're going to have the messenger RNA that we just copied from the nucleus, from the DNA. We're going to have the ribosome, which are the protein factories, the protein makers, right? And then we're going to have a second set of RNA, something called tRNA, also known as transfer RNA. So this is where the translation actually occurs. Remember, the messenger RNA is in all those letters that we just said, the UA, CC, UA, whatever. The tRNA is delivering the amino acids, your tryptophan, your isoleucine, your valine, all these things that build proteins. So we need that translation. We're going once again from Russian to Spanish. So the transfer RNA is going to help us with that translation. Well, I shouldn't draw the arrow. There we go. The messenger RNA going to the amino acids. And we'll learn how this codes on the next slide. So once again, happening in the cytoplasm. And how this works, how the transfer RNA actually brings it to the ribosome and how we actually make amino acids from this is it exists in something called codons. And another word for a codon, or another way of saying a codon, is a triplet. So everything is going to be in sets of 3 regarding the nucleotides, the messenger RNA, the amino acids. So remember that we have 20 different amino acids in the human body. Each codon, or each triplet, corresponds to 1 amino acid. So if I have a triplet of letters such as AUC, that's a triplet. That is 1 codon or 1 triplet. AUC codes for 1 of those 20 amino acids. Let's say tryptophan. Another, let's flip it. Let's say CUA. Now we're using the same 3 letters, but we're flipping it. It's completely backwards, and therefore it's going to be a completely different amino acid. So if you take a look at this picture, it's actually a really good illustration. What you can see here is UAC helps code for this specific amino acid. And then the next codon, or the next triplet, comes down here. Let's say AAA codes for this specific amino acid called phenylalanine. And then we just keep on going. It's just basically like an assembly line. So then we bring in the next triplet of the next codon, and that codes for something called arginine. And what we're doing is we're lining up all these amino acids. So in this picture, at the very bottom of the screen, if you can see my green arrow, there we go. What we have is we have one triplet, two triplets, three triplets, or three codons. So we have 9 total letters, or 3 codons. And what that's going to mean is it's going to code for 3 amino acids. So one codon codes for 1 amino acid. If I have 52 codons, I'm going to have 52 amino acids. And if you put you put 52 and 400 amino acids together, you're going to get your proteins. So that's what we're doing here. This is the translation part. We're taking all these weird letters, A, U, C, C, U, A, and we're translating them to amino acids, and we're lining them up. And the ribosome's lining them up and keeping them in order, and that's eventually what's going to lead to our protein. If you take a look at this slide, you can see that all 20 amino acids are present, and they all have their unique codes. So you can take a look at all of them. You're not going to have to memorize any of these by any means, but depending on what 3-letter order, AAA is going to be different than CGU, which is going to be different than ACC. So eventually, though, you put all those different orders together, they code for all 20 amino acids. Now, it's not just the coding which amino acids we have, it's also the order of amino acids that's important, that we talked about. So if you have a tyrosine and a tryptophan and another tyrosine, that's going to be very different versus if you have a tryptophan and then 2 tyrosines together. If the lineup or the orientation of them are different, that's going to code for a different protein. So the order does matter. And the ribosomes are in charge of making sure that order is all put together in the proper positioning. Okay, we've covered transcription, we've covered translation. We know how we make proteins. This last section of this chapter is cell division, cell growth, how we make new cells. And we've already covered this. You already know it, right? How do we make new cells? How do we make new skin, bone, nail, hair cells? We double the DNA. We're going to take our 46 chromosomes and double it to make 92, and then we're going to split them in half. And that's it. But now what we're going to do is we're just going to go in a little bit more detail and learn about the different steps of how this actually occurs. So what this whole process is called is the cell life cycle. The cycle of life, right? And the cell life cycle consists of 2 phases. Number 1 is called interphase. That's the first one. And then the second phase is called the M phase. And you can see the M could stand for 2 different things. It could stand for mitosis, if we're talking about bone cells, skin cells, pancreas cells, gut cells. And then it could also stand for meiosis, and that's going to be your sex cells. So that's going to be your sperm in guys and your eggs in ladies. So depending on what we're making more of, it can be mitosis or meiosis. But it's always going to be in this order. It's always going to be interphase first, followed by the M phase, if we are duplicating or making more cells. So what we're going to do on the next several slides are we're going to cover the subphases of these 2 phases. So big picture, interphase is first, M phase is second. And we're going to dive into the details of what's happening during interphase, and then the details of what's happening during the M phases. So take a look at this illustration on the right, and what you'll see is this is kind of our starting point right here at 1 o'clock. I'll put ST for start. And as we are making new cells, all of this in green is interphase. So you can see the majority of a cell's life is spent in interphase, or at least, sorry, life cycle is spent in interphase. And then this little tiny sliver, which would actually be the finish line or the end, that is called the M phase. So as you can see, the interphase portion has 3 subphases to it, and they're called G1, S, and G2. And on the next slides, we're going to be covering what G1, S, and G2 are, what's going on. But basically what we're doing is we're just getting ready for the cell to split in half. So we need to make the cell a lot bigger. We need to double a lot of things, and we also need to double all of that DNA. So that's what we're going to be doing during interphase. And then mitosis and meiosis, we're going to be splitting stuff. So interphase, growing, M phase, breaking things apart. So when we do break things apart, once again, there is a difference between mitosis and meiosis. First off, mitosis only happens in what we call somatic cells. And what somatic cells are is everything that is not a sperm and egg cell that's capable of reproducing. So as I keep mentioning, I keep giving you all these different examples. These are your lung cells, your pancreas cells, your kidney cells, your spleen cells, your skin cells. Everything that's not sperm and egg is referred to as a somatic cell. And for the most part, the majority of your somatic cells do go through mitosis all the time. And the result of mitosis is we're going to take one cell. So here you can see in this picture on the bottom, we got one cell. And eventually what we're going to get is 2 identical daughter cells. Now the reason why they're identical is because we are copying the DNA. Those 46 chromosomes are simply being copied and pasted. So they're the exact same. There is no difference. So when you burn your mouth on hot pizza, and it kind of blisters, well, not blisters over, but you have it, that weird scratchy feeling, and it's kind of numb or it kind of hurts for a few days. After a few days, when it heals, it's the exact same tissue, the exact same cells. It's not the exact cell because you killed the cell, but the cell is made up of the exact same DNA. And therefore, there's really no different than the one compared to the one that you killed. So that's mitosis. As I mentioned, with meiosis, this is going to be your sex cells, or what we call our gametes, is another way of saying sex cells, so sperm and egg. What we're going to be doing in meiosis is we're going to be doing basically mitosis twice. So we're going to take one cell, split it into 2, and then these 2 cells are going to split into 2. So what we're going to end up getting is 4 cells. 4 sperm cells or 4 egg cells. And what we call that's called haploid. Haploid means one set of DNA information. So remember, when we make babies, or you, if you look at yourself, we have 23 chromosomes from mom, 23 chromosomes from dad. So all of us have either sperm or eggs. And even if you're a hermaphrodite and you have both testes and ovaries, generally only one of them is actually functional. Only one actually works. So this is what we call haploid, meaning it's only 1/2 of the potential DNA information for a human being. So when we have 46 chromosomes in a cell, we call that diploid. And it's just, I'm going to put dip, D-I-P, and then loid, as you can see the spelling. And if we have 23 chromosomes in a human cell, such as sperm and egg, we call that haploid. So it's just half of the diploid. And we'll talk more about what's going on with meiosis at the very end of the chapter. So that's the difference between mitosis and meiosis. It's just where are the cells? Are we talking sperm and egg, or are we talking everything else? Once again, the cell life cycle consists of two big phases, big picture. So the first one is interphase, and then the second one is M phase. So let's talk about what's going on in interphase and the three subphases of interphase. As I mentioned previously, interphase is just a time frame where the cell can grow and multiply pretty much everything. So that's what we're doing. As you can see, what are we growing? We're growing everything. Everything that's in the cytoplasm. We're growing the nucleus. We're going to be making more DNA. And we're growing the plasma membrane. So those are the three structures of the cell, right? Plasma membrane, nucleus, cytoplasm. So what are we growing? We're growing everything. We're making more proteins, we're making more lipids, we're making more carbs. So we're making all three big macromolecules in our diet. And then with the synthesis of your DNA, that's our nucleic acid, so we're making all four macromolecules. That carbon, hydrogen, oxygen, nitrogen makes up all the four of these things. So very, very, very busy time. And this busy time can be separated into three subphases down here at the bottom. And in order, once again, they're called G1, S, and G2. So the S is in between the two G's. Now the G actually stands for gap, so it's gap 1 and gap 2. But you can really study it and remember it as growth, because that's what we're doing. So growth 1 and growth 2. So during G1, we are growing. We're growing our proteins, our carbs, our fats. We're growing our plasma membrane, cytoplasm. We're growing the number of organelles, our mitochondria, our lysosomes, Golgi apparatus. Everything's getting bigger. And then in the S phase stands for synthesis. And another way of saying DNA synthesis is DNA replication. So synthesis means to make, replication means we're copying. So how do you make new DNA? Well, you copy it, exactly. So we're copying our DNA here in the nucleus. And then once all the DNA is copied, then we're ready to grow a little bit more. We're going to make more proteins, more organelles, plasma membrane gets a little bigger, and everything just pretty much doubles, and then we're ready to split. So once again, big picture, why are we doing this? We're taking one cell and we're splitting it into two. And then if we're talking sperm and egg, we're eventually going to split it again into two more, right, for 4 total. But if we're talking, let's just say we want to make new skin, nice and simple. We need to take a skin cell, double it in size, double it in everything, and then we're going to split it into 2. So we double the amount of cells that we have. So yes, even though we kill millions and millions of cells per second, you're hopefully making millions and millions of cells per second to counteract that. So G1, S, G2. Grow, make DNA, grow some more is what we're doing. Then we get to the M phase. Let's talk about mitosis first, because that's the majority of your cells, right? The non-sex cells are somatic cells. And mitosis is, now that we've doubled everything, we've copied everything, we're ready to split everything. So the actual definition of mitosis is the splitting of the nucleus. There's going to be one more extra phase at the very, very end that helps us split everything else, splits the plasma membrane and the cytoplasm. But right now we're just focusing on splitting the DNA. So in the S phase of interphase, we doubled everything. We had 46 chromosomes, and we made another 46. So now we have 92 chromosomes, and we're going to split them. So 46 go to one cell, and 46 go to the other cell. And how we accomplish that is through 4 subphases. Just like we saw G1S and G2 in interphase, mitosis has their subphases. And they are as follows: prophase, metaphase, anaphase, and then telophase. So you do need to know the order of this. Once you understand what's going on in each phase, it'll be easy to put them in order. But a really good way to remember this is just the first letter of each word spells PMAT. So if you have a dog or a cat and it's not house trained well, I guess, and it pees on your rug or it pees on your mat, PMAT, you have mitosis. I know it's silly, I know it's weird, but you won't forget it on the test, right? If you remember PMAT, you know the order of mitosis. So let's talk about what's happening in each of these 4 phases. Once again, big picture, we're splitting the DNA. We're splitting the nuclear material. So here is prophase, first letter of our PMAT. And what's happening is we are getting ready to kind of organize the chromosomes. So we have 92 chromosomes. It's just in a big bundle, big mess. Remember, we have to kind of fold and wrinkle and spin and twirl and kind of compact all this stuff for it to fit inside of our nucleus, right? So what we're going to be doing is we're going to kind of condense the chromosomes. The nuclear envelope is going to start breaking down. Remember, the nuclear envelope is basically the plasma membranes of the nucleus. It's just the outer wall of the nucleus. And we're going to break that down because we're eventually going to split the nucleus. And what's going to happen is these spindles, these proteins are going to start forming. And the spindles are kind of like these little legs, or these tentacles, if you will. And what's going to happen is these tentacles are going to grab on to the chromosomes, and they're going to pull them in different directions. So we're going to have half the chromosomes getting pulled by the tentacles to the North Pole, and then we're going to have the other half being pulled to the South Pole. And at the end of it, we're going to have 46 chromosomes on each pole, North and South Pole, total opposite ends. So prophase, we're just kind of organizing things, condensing things, lining it all up. It looks like a big bundle of spaghetti, kind of. You take a look at this picture here. The blue thing, this blue circle, is the nucleus, and we're just going to kind of just get the chromosomes all ready. And what's going to happen is we're going to be actually moving them on the next phase. And then this yellow gold stuff around the nucleus, those are the spindles that I mentioned. They're kind of the octopus legs that are going to be grabbing onto these chromosomes in the next couple phases. So PMAT, P is prophase. The next phase is metaphase. And in metaphase, what we're doing, you can see the picture looks very different now. You can actually see some organization here. What we're doing is we're lining the chromosomes up, which are these blue things that I'm circling in red. We're lining them up in the middle. So the way I always remembered it was M for metaphase, M for middle. So they kind of align along this imaginary line called the metaphase plate, or the equator, if you will. You can see that we're forming a north pole down here and a south pole over here. And that's really all that's happening. We're just lining everything up. Because in the next phase, that's when they're going to start to rip apart. They're going to be pulled apart. So M for metaphase, M for middle. Anaphase is our next phase in the PMAT. And what's happening here is, as you can see in the picture, the chromosomes are being ripped apart. The previous slide showed that they were all lined up nice and neat in the middle, and now they're being separated. So I always remember this A for anaphase, A for apart. Or I guess you can say A for away. So at this point, they're not chromosomes, they're called chromatids, where the terminology changes during this process, but basically the genetic material actually gets split apart. So what we're eventually going to get, our end game product, is we're going to get one cell here with 46 chromosomes, and then we're going to get another cell here with 46 as well. So those spindles that were starting to form during prophase, that's what those little octopus legs, that's what's actually pulling the chromosomes apart towards their respective end of the cell. Okay, last but not least is telophase with PMAT. Now, if you take a look at this picture, it's still one cell. they're not totally disconnected yet, even though they're pretty close, right? You can see two big circles, but they're still connected by cytoplasm. So technically this is still one cell. But if you notice, the nuclei are totally separate. We have one nucleus in this cell and one nucleus in this cell. So the 46 chromosomes have successfully made their journey to each end of the cell, and we now have two distinct nuclei. So we start reforming that nuclear envelope and the nucleolus and everything, and now we're pretty much ready to just split the rest of the cell. But remember, our definition of mitosis is splitting of the nucleus, splitting of the DNA. So that's PMAT. In order to split the cytoplasm, we have to go through another process called cytokinesis. And cyto means cell, kinesis means to move. So we're moving the cell to the point where it actually splits. Now, a lot of times telophase and cytokinesis happen simultaneously. They're happening at the same time. But every once in a while, these things will get stuck and they'll just exist in telophase. But once the cytoplasm continues to be pulled towards their respective cells, this little indentation called the cleavage furrow will actually pinch off, and what we'll have is two identical daughter cells. Exact same DNA, exact same organelles, exact same everything, but they're two cells now instead of one. So here you can see cytokinesis. Once again, it means movement of the cell, so specifically cytoplasm. And once PMAT is completed, or at least once PMA is completed, we can start making this indentation more significant until the point where they just kind of pinch off. And then once again, our end result is two brand new identical daughter cells, same DNA. So let's recap this. You just covered the cell life cycle. The two big phases are interphase and the M phase, but both of those have their own subphases to them. So if we're going to put this all together by subphases, now you know the order of this. So I'll do this on the bottom of the slide. First thing to happen in interphase is G1, gap 1, which we said growth 1, right? We're growing everything. Then we have the S phase, which we're synthesizing DNA, copying, replicating DNA. And then we have our G2. We're growing some more. So that's interphase. Then we go into mitosis, which is our PMAT. We have prophase, metaphase, anaphase, telophase. And then last but not least, we have to split the rest of the cell. So PMAT just splits the nucleus. We have to split the rest of the cell, and that's called cytokinesis. So G1S, G2, PMAT, CK, now you know the whole order from beginning to end of how a cell makes two identical daughter cells. So this is a great review slide to show you where you're at. You can see how this is kind of a combination of interphase and prophase. And then you can see where the chromosomes are lined up in the middle here would be metaphase. So this is just kind of a transition phase between the two. Here you can see on the next one, you can see that they're being pulled apart. So A for anaphase, A for apart. And then transitioning to them being pulled apart, you eventually have telophase. And telophase, remember, it's still one cell, but you have two nuclei. And then we can see cytokinesis just pinches them off, so we have two identical cells. So last topic we have to cover is the difference between meiosis and mitosis. And essentially what we're doing with meiosis is we're just doing mitosis twice. So as you can see, we're going to start with interphase, And we're going to start with one cell. And then what we're going to do is we're going to go through mitosis, which we call meiosis 1, which gives us 2 cells. And then we're going to do the whole thing again. So we're going to do PMAT once to get 2 cells, and then we're going to do PMAT again to get 4 cells. So remember, meiosis is only occurring in the sex cells, in the gametes, once again, sperm and egg. And what we're doing is we are actually shrinking the amount of chromosomes, because this is only unique to guys respectively and girls respectively. So we're taking our cells, our 46 chromosomes, we're doubling the DNA to make 92, but then we're splitting them to 46, and then we're splitting them again to make our haploid number, which is 23. So in a guy's sperm, there's only 23 chromosomes. In a lady's egg, there's only 23 chromosomes. When they get together, the birds and the bees, magically, you get a baby, and that baby hopefully has all 46 chromosomes in their gene pool, in their DNA. So, let's go on to the next slide, talk about meiosis 1 and how this differs just slightly from mitosis. So with meiosis 1, we're going to be going through PMAT. It's the same thing. We're going to be going through prophase, metaphase, anaphase, telophase. But what's unique to meiosis is during prophase 1, some genetic shuffling starts occurring. So we kind of get this movement of the chromosomes or of the genetic material, and they call that crossing over. So what happens is, if you take a look at the end product, what we're going to get is we're going to get variability in these 4 cells. They are not going to be identical. So you can see in this top cell, we have a lot of red on this chromosome on the left and not a lot of red on the one on the right. And then you look at the second one, it's all red. You look at the third one, they're all different, right? And that's why brothers and sisters are different. If you're not an identical twin, even if you have the same mom and the same dad, there is genetic variability because only one sperm makes it to fertilize the egg, and there's only one egg that makes it, that's able to be fertilized by, that's ovulated and is able to be fertilized by the sperm. So even though you have the same mom, same dad with the same genetic material, there is a lot of variability. So if you have 2 kids, 5 kids, or 9 kids, it doesn't matter. All of them are going to be a little different. They're all going to have characteristics of mom and dad, but they're not going to have the same characteristics or the same distribution of them because of this phenomenon. And then in meiosis 2, we're just splitting everything to get to that 4. So basically, remember, we're starting with 1 cell, just like we did in mitosis. We go through meiosis 1, and the result of that is we get 2 cells that are a little different, and then those 2 cells split. And we end up getting 4 different cells. They are not genetically identical. So mitosis, they were the same. Meiosis, they are not. Which is kind of a good thing, right? We don't want brothers and sisters and cousins. We don't want everybody looking the same and being the exact same. That would be kind of boring, right? We want everybody to be a little different. All right, everyone. Done. A little over 3 hours, but remember, in a 16-week semester class, you're supposed to meet for 3 hours a week. In an 8-week class, you meet close to 3 hours a week. So this is I'm 2 minutes over, but this is on par with what you would get from a face-to-face class. So, hope you enjoyed it, and now we're going to move on to Module 2. So we put our chemicals together to build cells, and our cells are going to be put together to build tissues, and that's going to be our next topic. So I'll see you next time.